America, course coordinator Dr. Mohammad Zain Khan, assistant professor at Department of Industrial Chemistry, Aligarh Muslim University. My dear students, Assalamu alaikum and a very good morning. Today, on the last day of this 5-day GIAN program, I welcome you all. And in the morning session, we'll be having a very nice and meaningful and very interactive type of session with Professor Liu. So I urge all my students to get involved. So with this, I invite Professor Hitao for the lecture. Thank you.
Thank you for the kind introduction. Okay, this should make the Zoom to work now. Okay, all right. Thank you for coming back. I understand today's a holiday, so I appreciate students and faculty giving up the precious personal time attending this sequence of lectures. Today I'm gonna, well, in this lecture I'm gonna provide overview and introduction of DNA nanomaterial. This is a new emerging and rapidly developing area of research. So my talk in the morning would be more on the general overview, and we'll spend more time on the manufacturing, fabrication of those nanostructures, the chemistry principles behind those construction of those structures. I will briefly touch on the applications, focusing on the unique properties of this class of materials. In the afternoon, I will, well, the lecture after this one, I will focus more on my own research program in this area, in particular using DNA for large scale and high resolution nano fabrication. And in the afternoon, I will be able to show you some videos recorded in my own lab, showing how students in my lab are making those DNA structures, but also characterizing them using atomic force microscope. We have some technical challenge. Maybe just show the PPT first or video? Just show the PowerPoint. When I show the video, I'll just switch back. Good. All right. Hopefully you can, colleagues online can see the shared screen now. Okay, so if you missed my previous lectures, this is where I come from. I am a faculty member at the University of Pittsburgh Department of Chemistry. This is located in the eastern part of the United States. Pittsburgh is it's the second largest city in the state of Pennsylvania. Let's see, pointer options. And it's located in the western part of the state. So before I jump into the topic, I want to do a little bit of flashback and give you a little bit of context here compared to the nanomaterials that I talk about in the previous days, but also you probably are very familiar with. And I also was asked a number of questions about different forms of nanomaterial. So I want to briefly talk about why DNA and how DNA nanomaterial is different from many other nanomaterials. So quantum dots or nanoparticles, either metallic, if it's metallic or oxide, usually people just call them nanoparticles. If it's semiconducting, often semiconductor nanoparticles are often called quantum dots. And the reason being, those are small small particles with their size around 2 to 100 nanometer. Now, why semiconductor nanoparticles are also called quantum dots? Because there's a phenomena called quantum confinement. You have a wave function has finite size. In a bulk material, the size of the material would be greater than the size of a lot of the wave functions you are dealing with. In that case, the material property is not limited by the material itself. Okay? Now, when you have a much smaller particle, thinking about 2 nanometer particle, but the wave function in some cases can expand 2, 3, or even 4 nanometers. Now you come to this interesting geometry and size regime where the size of the material would impose a limitation property. And you can actually change material property by simply change the size. In another way of seeing this is that there's a quantum confinement in the nanoparticle if the size is right. And that quantum confinement is happening in all the 3 dimensions, x, y, and z. Another way of looking at this is a 0-dimension material, meaning that all 3 dimensions you have a quantum confinement, and there's no dimension that is not quantum confined. So that is where the 0 dimension comes from. We also talked about carbon nanotubes a couple of days back. Now those are tubular structure. If you think about material in terms of geometry, you can view this as quantum confinement in 2 dimensions. Y, but not Z. If you think about carbon nanotube along in the Z direction. So wave function propagate in the Z direction, but confined in the X, Y. And again, this will produce a modification of electronic structure and band structure compared to the bulk electronic structure. So in the case of quantum dots, light emission has been a very popular application. You may have seen or terminology like this. And what is happening is they actually put cadmium selenide quantum dots in their display, because those quantum dots can produce very pure color, very pure red, very pure green, and very pure blue. So those 3 colors will reproduce all the possible colors out there. In the case of carbon nanotube, there's a similar property in terms of modulation of emission, but most of the applications are in electronics where semiconducting properties are being taken advantage of. Being one-dimensional also creates additional benefits in terms of making device, because a lot of devices, well, pretty much all the devices have finite size. So when you're dealing with small particles, you have to think about junctions between particles. If you're dealing with a tube, then there's no such junction you have to think about. So that makes it easier and more suitable for manufacturing nanoelectronic devices. Now it goes to graphene 2D materials. Those are materials having very narrow thickness, but in the X and Y direction, they can be infinitely large. Okay? So those are materials having quantum confinement only in 1 dimension. So instead, they're called 2D materials. 3 minus 2 is 1. Okay? So 1 dimension of quantum confinement, and the other 2 dimensions are not quantum confined, and they're called 2D. In the carbon nanotube, it's 1D, meaning that the other 2 dimensions are quantum confined. In the case of quantum dot, all 3 dimensions are quantum confined, leaving them a name of zero-dimension material. Now, those materials are all very interesting in the sense that material properties can be a function of size. Okay. The DNA material I'm going to talk about today is a little bit different. In this case, the basic building block of the material is double-stranded DNA. This is the foundation of life. If you think about the double helical structure, it's about 2 nanometer in diameter. Okay, you can view it as a wire, essentially. What I'm going to show you later is how you can start from this very simple wire structure to construct higher order structures as large as several hundred nanometer in dimension. Now, this class of material is covalent in nature. It's covalent, but also there's certainly covalent linkage within the DNA chain, but also there is non-covalent interaction, hydrogen bonding, electrostatic, that pull the chain together. There's no quantum confinement in this case because DNA is pretty much an insulator by most people's standard. So we're not looking at excited state dynamics. We're not looking at many things that we typically look for in a quantum dot, carbon nanotube, or 2D semiconducting 2D material regime. There's no light emission, at least not very strong light emission. Instead, we're looking at many other properties that are fascinating, in particular, the exquisite control over the size and the ability to manipulate the location of different material components. So if I have to summarize this slide, there's a big difference in terms of motivation, why we want to study those material, and what are the unique strengths of each kind of material. Okay, so all this business happened about 40 years ago. The person on the left, his name is Nat Seaman. He was a professor at New York University, chemistry department. And he's considered by almost everybody in the field to be the father of DNA nanotechnology. He actually has a PhD degree from University of Pittsburgh. So he's actually our chemistry department outstanding alumni awardee. And he was trained as an X-ray crystallographer. So if you recall the history of United States, 50, 100 years ago, there's heavy industry, steel manufacturing, heavy metal processing. So Pennsylvania, being an industrial harbor at that time, has a department of crystallography. Okay, so students studying how to do single X-ray, structure, refinement, things like this. So he got a degree in doing X-ray diffraction. Now that department later merged into chemistry department, which makes us consider him being our alumni. Now he's actually he's not a DNA person, but rather his interest is to think about resolving protein structure, and to do that you have to grow protein single crystal. And he started his independent career in NYU, New York University, around late 1970 or early 1980s. And he spent his time resolving different kinds of protein structure, basically. But it's frustrating. I don't know if you have done those, but proteins are big molecules. It's difficult to crystallize them, very difficult. So he would be frustrated not being able to get protein crystal, can't figure out how to do them. If he doesn't have a crystal, then he cannot do X-ray study. So, and one day, this is a story told by him, and one day he went to a bar near the university campus. So he is thinking about a research problem, how to make a crystal of protein, because X-ray happens only when you have regular array of materials, right? You have to have this lattice. But growing protein structure into a single crystal is difficult. Now, how can he get around this problem? It turns out, well, just as fate may have it, in the bar there's this art, the one hanging on the right. I think it's called something like The Depth or something like this. I'm not an artist, so that name of the picture escaped me. But it's a pretty well-known art piece produced in the 1950s. And it shows this array of fish. Okay? And the array extends in 3 dimensions. And each fish has this fin in in 2 dimensions, and they're somehow aligned together. Okay, so to the eyes of an X-ray person, this is a crystal. You have 3-dimensional repetition. And suddenly, somehow, lightning strikes him, whatever happened. And he started to think, oh gosh, how can I make artificial lattice, a 3-dimensional lattice? If I can do that, can I just attach a protein to each of the units? And if I can do that, then I will have a way to position protein in a regular lattice structure. Then I would have a crystal. I would have a crystal not dependent on the particular shape of the protein, but rather dependent on extra sets of framework. So just thinking about all the scaffold you encounter in a building a high-rise building, you can always make this large terrace structure, and if you can put molecules at exactly the same location within each unit cell, then you can create an artificial crystal. So this became the starting point of a wave of evolution in structural DNA nanotechnology. And this very simple idea of using something to create a molecular lattice framework become the underlying principle and thought and philosophy of DNA nanotechnology. So after that, he just started thinking about how he can use double-strand DNA to create this lattice structure, and how he can then start from there, attach different molecules onto this DNA lattice, and then from there, a lot of other things happened. Turned the research into something he never imagined, a very productive research area. Unfortunately, he passed away 4 years ago. Before he could have won Nobel Prize, in my view, if he lived another 20, 30 years, probably he will won another Nobel Prize in chemistry. All right, so looking at the very fundamental structures, now this is a little bit more biochemistry. What I've shown here on the bottom is the bottom right is the conceptual idea that Nassim had in the 1984, I believe. So here, I show this lattice structure XYZ, and here's this some kind of blob, greenish, bluish blob. This is the sort of the protein that I want to attach to the lattice. If you can attach it to the same space, same area, same location, then you can come up with some kind of cubic lattice of protein. Okay. Now, how can you do this? Well, what Ned Simon thought about very early on was some kind of junction, a DNA junction, and one type of junction he worked with in particular was so-called holiday junction. And what this is essentially four strands of DNA come together. And you look at the structure here. This is the schematics. You have 4 DNA strands, and each DNA strand can be broken into 2 segments. And each segment is recognized by another segment of another DNA strand. So if you have a structure like this, you can essentially combine, I'll reject. You can combine 2 double helical structures together. So essentially, you are threading 2 wires into one. Okay, that's the way I look at it. Now, if you have, and the way you can draw them is just like a cross. Okay. But another way of looking at it is you have double helical structure on one side, another double helical structure on the other, and in the middle you have two crossover. Okay, the two in the middle is a crossover structure, and this crossover structure brings the hydrogen bonding structure together. So instead of have one wire, you have two mechanically interlocked together. Now you can design this structure to have multiple crossover. You can also design structure to have so-called sticky ends. A sticky end is elongation of, is shown here. So if you look at this motif, one end of the, one part of the double helical structure is elongated a little bit longer. And this produced a little bit of additional single strand motif. Now this one will be free to hydrogen bond to other parts of single strand DNA. Okay. Now imagine if you have a lot of those things, you can create, those are essentially cross structures, but can polymerize onto themselves. As long as you design the end of the sticky ends in a particular way to recognize themselves, then you can polymerize this in 2 dimension or in 3 dimension, so on and so forth. And once you can do that, you would have a 3-dimensional, 2-dimensional DNA lattice. Okay. So this is a very rough idea that Nate Simon put forward in the mid-1980s. Once people got into this, concept, things become things start to evolve rapidly. So here, what I show a so-called tile-based structure. Why I call it tile? If you look at this this crossover, and this crossover is another upgrade of what I show here. Here you have one crossover, right? But you can have multiple crossover within the same sequence. Well, within the same hybridization, I guess. So here, for example, you have you can view it as two double helix DNA in parallel, but it has one crossover here and another one here. There's actually there's a circular one here as well, and this is called DX. You have 2 become 1, and you can do it over even larger lateral dimension. Imagine you are making a carpet from threads. You put a threads in parallel and somehow connect them together. This is exactly what you're doing, except you're doing it using molecular scale wire and a thread. So once you do this, you can make a larger structure, and you can play with different kind of crossover. Instead of having an individual, you could have 2 together, something like this, and this can improve the mechanical property of the nanostructure. So using this concept, you can using them as a building block to create much more complex structures. And I show some examples here. This is a small building block produced by multiple crossovers. And also, at the end of the building block, again, you see this small niche, small hangover, just 2 bases long. Okay. Now, the sequence here is designed in such a way that they will find themselves and polymerize themselves in 3 dimensions, X, Y, and Z. Once you have a lot of the structure in solution, and this is the X-ray structure of the unit, they can, as I said, they can polymerize in X, Y, and Z and actually produce crystals. Okay. And shown here is an optical photograph of the DNA crystal, a porous DNA crystal being produced using this technique. This is exactly what Nassim envisioned in 1984 or 85. Took the community about 30 years to make it work. You can also, using this technique, instead of folding it, instead of polymerizing onto themselves, in that case you get a lattice, X, Y, Z dimension lattice. You can also have the structure to recognize another portion of itself. So if you do that, here's one example. You can use this fundamental building block to create intermediate scaffold. And in the intermediate scaffold would recognize either themselves or with another scaffold. And by doing that, you can create polyhedral structures. And those would be on the order of several nanometers or several tens of nanometers. It's a very different way of making and designing DNA structure of pretty much arbitrary shape. So what I showed in this slide is some kind of repetition lattice. That's interesting, but what if you want to write your name using DNA? That's much difficult to design here, right? Because here you have a lot of constraint that you have to work with, and there's no way of scaling this up easily. This scientist at Caltech, his name is Paul Rothman. He developed a very different idea, and his concept is to taking the hint from origami, which is the folding of paper, and he took it into the nanoscale world and thinking about folding of the DNA strand. So on the top left is the concept of this DNA origami approach. And what I showed there is a lot of wires. There's a black line that goes through. So there's a black line, and you can imagine this is a very long single-strand DNA. Okay. And there are bacteriophage DNA that are several thousand bases long, and this is one of those that you can use. And this strand is called, in the community, called scaffold strand, because it is actually being used as a scaffold of the construction of a DNA structure. What is also present are a lot of colored lines. Now, those color lines, those are also DNA strands. They are much shorter in sequence. Usually, they're about 20 to 40 bases long. Okay. But there are a lot of them. And I also want to emphasize that each one of them is unique in sequence. Okay. Now, what is happening here is that you have a very long DNA strand, and you know the sequence, you know the whole sequence of that long DNA strand. Okay. Think about 7,000 bases long. You can create a lot of staple strands, those short strands, each being about 30 to 40 bases long, but you make a lot of them. In reality, you make about 300 of them. So 300 DNA strands, each one being unique. What you can do is you can design the sequence of a staple strand such that two portions of the staple strand would recognize two portions of the scaffold strand. But those two portions on the scaffold strand can be really far apart. Okay. Now imagine what's going to happen when you mix the two together. The DNA would perform hydrogen bonding. As I said, the two portions of a short strand would recognize another two portions of the long strand, but because the two portions of the long strand are so far away, what's happening is the short strand essentially burn the two parts of the long strand together. Now, essentially what is happening is you are folding the long DNA strand by using a short DNA strand, the staple strand. So you're stapling the long DNA by folding them in half. What is powerful in this concept is because there are a lot of short strands, as I said, about 300, you can apply this concept in an iterative fashion. Essentially, you can fold the long strand 300 times. And this is exactly what is happening here. Okay. By folding it 300 times, you can start with a very long wire, seemingly having no structure, but you can fold it in any way you want. You can fold it any way you want and fold it 300 times. Imagine if you have a piece of paper, you fold it 300 times, you can produce any shape you want. Right? Same thing happens when you fold a piece of wire, except in this case, you got a two-dimensional shape instead of a three-dimensional shape when you do actual paper origami. So if you look at the pictures here, okay, the pictures here shown are models of DNA structure, and on the bottom are actual structures produced by this technique. Each one of the structure is about 100 nanometer in size. Okay. To give you a context, your hair is a couple of micrometer. So this is about 1,000 to 10,000 times smaller than the diameter of your hair. Now, even with that kind of small dimension, you see very exquisite control over the shape. And this smiley face being one prime example, you can essentially fold it into any shape you want within that limit of 100 nanometer by 100 nanometer size regime. Using the same kind of approach, you can also create a tile. So the B panel shows a very similar origami approach, except it's a little bit connected, a bit more connected to the tile-based structure. Here, what they did is they create this regular tile, and then they fold the tile into defined pattern. And this is more like a piece of paper being folded back and forth, back and forth. But the same kind of idea. You use short strands to recognize two portions of another structure and fold them into geometric shapes. If you look at the bottom, you see that this kind of concept can also be applied to make three-dimensional structure. On the bottom, there's some kind of ring structure that mimics a gear, a mechanical gear, except is probably the smallest gear you can make with the size of around 100 nanometers. On the right-hand side, it's even more interesting. This is a nanoscale flask. You can fold everything into an irregular shape in the 3-dimension, and this whole thing is about 40 nanometer in this dimension. So a very exquisite control of the folding of very flexible material, and that is in my view, the true evolution of DNA nanotechnology. It really enables people to make arbitrary shaped patterns on both 2D and 3D. To summarize this slide, I want to recap the two main components. One is a scaffold strand. This again is a very long single strand DNA. And one of the most often used one is called MP13. That is a bacteriophage DNA about 7,000 bases long. And then what you need is a lot of short staple strands, depending on the size of the nanostructure. The typical structure made here requires about 300 unique DNA strands. Okay. Again, each one is unique. Okay, I'll pause here for a bit and to see if people have any questions on the construction. Are you talking about the fold sequence? Yeah. Okay, that's a very good question. So if you think about this kinetic process here, you have one long strand DNA and 300 different strands. In reality, what happened, when we make them, in the afternoon, I'll show a video of this. We actually mix all 300 strands and the scaffold strand together. And we don't even care too much about the ratio of them. And we heat it up and cool it down, and magically the structure is being produced. So if you think about this being a stepwise process, it feels almost impossible because there are so many different ways this could go wrong. And indeed, there is a lot of different ways this could go wrong. But at the same time, this is probably more a collective and self-catalyzing in some way. Self-catalyzing is probably the wrong word. But there are many different pathways, and it doesn't have to be folded by a particular sequence, but rather any one of the 300 strands can fold the long strand, and they just go through different pathways, but the final structure energetically is so favored, so it always go back to that location. And also, this would require very slow annealing So we typically anneal the reaction mixture for 10 hours or 20 hours, very slow cooling. So, so the structure has a lot of opportunity to fix the problem. There's dynamic equilibrium between hybridization and non-hybridization. If the structure got stuck, it can always go back and find another pathway to eventually go to the end. So it doesn't happen in every case that it will produce high yield, but Rothman's original approach is very easy to reproduce. My lab has no experience of DNA technology. When we started doing this, we reproduced their work in our second try. Any other questions on this? Okay. You can always ask the question at the end. All right. So the origami approach has been very popular and very powerful. But people also started to think about different ways of making the structure. And if you think about the origami approach, one of the biggest limitation is actually the scaffold. Because you need this very long single-strand DNA, and the length of that DNA actually limits the size of the structure you can make. Okay. But also, in addition to that, because the folding is, if you fold a square, you can just do this, right? If you fold a triangle, you have to do this. Okay. So the folding sequence would be very different depending on what kind of shape you make. So to make each one of those shapes, you have to make a separate set of staple strands. So what that means is if you want to make smiley face, you need to go to the DNA synthesizer company and buy 300 strands. Okay. If another day you want to make a triangle, you have to buy another 300 sets, and those 300 sets are totally different. Okay. Now, fortunately, there's a computer program to design everything, so you don't have to, work on paper and pencil to figure out what sequence you order. You can just put your shape into the program. Program will tell you what sequence to order. You can just order them. But that still is very inefficient, both from a material utilization, but also a compatibility across different shapes. So about 10 years ago, there's another approach being reported. This is called single-stranded tile approach. And it's also being referred to as DNA break. It all depends on the context you're using this approach. Now, this approach has the most significant feature of not using a scaffold. So in this case, everything, all the structures are produced by using a mixture of short DNA strands. Okay. And each DNA strand is the same length, 32 base pairs long. And this is one example. Okay. Imagine what you have here is a 32 base long single-strand DNA. You can artificially break it into 4 domains, okay. And each domain would be 8 bases long. Okay. What you can do is you can make, let's say, 10,000 of those strands. Again, each strand would be different in its sequence. But what you can, in the design phase, you can make the sequence only recognize its neighbor. If you look at this plot, this plot shows a bunch of single-strand DNA. Again, each one has 4 domains. Okay. If each one has 4 domains, you can think about in the XY dimension, each DNA sequence can recognize 4 nearest neighbors by using each of the 4 domains. Okay. If you can do that, then by designing the sequence, you can have DNA sequence 1 and 2 sit next to each other in this direction, and sitting next to each other in the vertical direction with different neighbors. Okay, so that's the idea here. You have 2.1, 2.2, 2.3, and it's designed in a way that that each one of them knows where they should be, because only if they are at the right place, their bases can hydrogen bond with other neighbors. If they're in the wrong place, then they cannot hadron bond with all their neighbors. So in a sense, it's like 1,000 students come to a big room, and you don't tell them where to sit. Okay, let's say we have 100 people here. Let's say, and I'm not going to tell you where to sit, but I put everybody into a room. I only tell you you need to sit next to your best friend A and B in this row. On your left should be A, on your right should be B. And in your front should be C, and in your back should be D. I'm going to tell everybody that information, and I'm just going to let you to figure out. And eventually, you will figure out how you should sit together. And the information I tell you would be the sequence information of the 4 domains. So by encoding that information in a specific way, I can let the DNA sequence know where they should be next to which sequence. Okay, so that's the idea. Now imagine if you have, let's say, 100 of those sequences, you can make them into 10 by 10 array. And that would be just a canvas, a piece of paper. You can make a flat, square-shaped nanostructure. And that would be something like this. Okay. Again, in this structure, each of the component knows where to find itself within this bigger structure. Now, what is more interesting now is if you eliminate some of those strands. Okay? If you eliminate some of those strands, for example, if I look at the structure and I'm going to draw a line and I say I'm going to get rid of all the strands on the top left, but I still keep all the strands on the bottom right in the annealing process. So in that case, the annealing will still happen, except it will only happen to produce a triangle shape. You can also say, I'm going to get rid of the strand that's supposed to be in the middle. And you do the annealing without that strand, you're still going to produce this rectangle structure, except in the middle would be a hole. Okay. Now, if you are being strategic, you can create shapes like all the alphabets, all the numbers, and all you need to do is to simply eliminate some of the strands in this tile. Okay. And by doing that, you can create 2D geometric shapes of anything. That is, the only limitation would be the size of the molecular canvas. Okay. And what's beautiful in this construction is that you can use the same batch of DNA sequence to produce any shape you want within that size limitation. You no longer have to design another 300 sequence every time you want a new shape. So all the shapes here, for example, are produced by the same batch of DNA sequence. The only difference is which one you are eliminating and which one you are kept in the annealing sequence. Now, this idea can be extended to 3-dimensional, and the only change you need to do is you need to create another dimension. So here, the 4 domains would recognize each other, but only within the same XY plane. What you can do is to play a little bit a trick to make the DNA sequence turn 90 degrees after the hybridization. So once you do that, you will be able to create 3-dimensional shape. And this is the same kind of idea. A lot of short sequences, they know exactly where they need to be in a 3-dimensional assembly. If you remove some of them, you can create geometric shape. If you include all of them, you're going to get a cube. Okay. So on the bottom are the actual shapes produced by using this DNA break approach. And those are transmission electron microscopy images. On the top are the models, cartoon models, and you see that the actual device mimics the model in a with very high definition. All right, this approach can be extended into even larger scale. If you design sequence in a way that it can polymerize onto itself, you can also create large-scale lattice structure. And here I show a couple of examples. This one where you have a cross lattice, this is still more or less 2D in nature. Okay. But this one, for example, you have a very large plate. Okay. And on this plate, you have a lot of holes. And this kind of structure turns out to be very useful in making high-density patterns for nanoelectronics application. Okay. All right. So the structures I talked about so far, the DN origami, their size limitation is about 100 by 100. And the single-stranded tile and DN break also has similar limitation because if you think about this, each domain here has 8 bases. Okay. Now, how many unique sequences can you get by using only 8 bases? Not many, because there's AT and GC. There are only 4 DNA strands. Right? If you do the math, you end up with thousands, maybe tens of thousands, somewhere there. My math is not very good. But we're talking about 2 to the 8th power or something to that order. So the number of sequences you can play with here is not that much. And once you start to have duplicated DNA strands, then the selectivity become a problem. Okay. So there are 2 approaches to solve this challenge. One is by doing stepwise assembly. This work was published by a group, again, at Caltech, where they produced, you may recognize this is Mona Lisa, except it's at the molecular scale. And what they did is they start with a small origami structure. Okay. And they designed a system where each smaller tile can recognize its nearest neighbor. So it's a combination of the origami and the single-stranded tile approach, except each individual building block become a DNA nanostructure itself. Okay. They were able to produce, by stepwise annealing of those components. They start with 2 by 2, and then 4 by 4, and then 8 by 8. So progressively building larger and larger structures. And this is an atomic force microscope image showing the pattern of Mona Lisa on the surface. So there's a video this group of authors produced. And let me see if I can show this video. I also see a chat. Can you see my video online? Good. Okay. Should I? Supposedly there should be audio. Okay, I can hear a little.
Repeated at different scales, taking. Nature is remarkably good at assembling complex structures such as human bodies or trees using simple building blocks such as cells. One of the principles that allow complex structural formation is called fractals, for example, as seen in this jacaranda leaf. A jacaranda leaf has nearly the same branching rules repeated at different scales. Taking inspiration from nature, we developed a strategy for creating complex DNA nanostructures by applying the same assembly rules repeated at different scales. We started with a square DNA origami tile. We designed how the tiles should interact with each other in a hierarchical, multi-stage self-assembly process. Molecules, in our case, double helical DNA, can be selectively attached to specific locations on the surface of each square DNA origami tile, creating a raised pattern that can be seen by atomic force microscopy. When all tiles are assembled together, from 64 test tubes to 16, and then to 4, and finally to 1 test tube, any complex pattern can be created, such as the Mona Lisa. The fractal assembly approach allows the same DNA strands to be reused for interactions between tiles at different stages of the assembly process. Thus, structures with increasing sizes can be created with the same small set of DNA strands. We developed an online software tool that converts the user's desired image to DNA strands and wet lab protocols. The structures that we created are 64 times larger than the original smiley face DNA origami and are similar to the size of a small bacterium.
Okay, so that this slide shows all the structures created by this group of authors, and as you can see, this is a excellent degree of control. Excellent degree of control over the molecular scale morphology and placement of materials over about 800 nanometer laterally. So this is a very impressive engineering feat. Okay, so I want to go back to my slides. Okay. So in addition to let me hide this. So another approach is simply have more strands. If you recall the two approaches, the origami approach always required this scaffold strand. Now if you go to the DNA break approach, that restriction is no longer there. And the restriction instead becomes the size of each domain. So if you make each domain longer, so the original size is 32 base pair. If you make it 52, then that becomes 13 bases for each domain. And with that, you can create as many as 10,000 unique single-strand DNA sequence. And what that means is you can put those 10,000 bases into a annealing solution and have them create larger and larger structures. And this is what is shown here. This group of scientists at Harvard, they created super large DNA brick structure, the largest being the cube. But if you intentionally eliminate some of them, you can create any 3D shape of your choice. So this shows the actual structure. Here the scale bar, I think, is 100 nanometer. So what you see here is a cube structure, not a 2D, but 3D. So it's 100 by 100 by 100 nanometer cube. There's this enormous amount of material packed into this nanoscale material, but also at the same time, within each individual component can be precisely controlled to the single strand level. you can add or remove one of the strands. And if you do that, you can create cavity, you can create three-dimensional shape, and those are the structures people have made, either as a positive tone or negative tone three-dimensional shape. It's like a sculpture, but using molecular additive components instead of microscopic material. Okay, so that's the discussion on the fabrication. I have about 15 minutes. I want to quickly go through the applications. The application, as I said, is different from the inorganic nanomaterials I focused on. Here, the unique feature is the ability to control the shape. Control the shape and control the positioning of components onto those nanostructure. A lot of the applications are involve some kind of patterning, and I'll spend a bit more time discussing the work in my lab that evolves around patterning. But also assembly of other materials. The assembly can be a molecular species, can be a nanomaterial assembly, can be biomolecule assembly, can be carbon nanotube, can be a lot of things. There's also a lot of research done in medical-related application. I'm not expert in this area, but I'll try to mention some of the interesting examples I saw in the literature. Okay, so on the assembly, there's this is probably the earliest application of DNA nanotechnology. A lot of the early work are actually not using a DNA nanostructure per se, but simply rely on strands. So those are some of the examples in the literature you find in the early days of combining DNA with nanomaterial. And usually you simply modify a nanoparticle. This can be gold or quantum dot, a combination of them. And if the DNA sequence are complementary, then you can essentially make the nanoparticle being able to recognize each other or recognizing a different kind of nanoparticle. So what you're doing essentially is you encode personality into a nanomaterial that otherwise does not have any unique name or feature or whatever that might be. With the mediation of the DNA strand, the nanoparticle can assemble into large-scale crystal in some cases, but certainly even at nanoscale, you can create structures like, large and small come together, things like this. With the DNA nanostructure in place, you have even more powerful controls you can apply. Again, because in every DNA nanostructure, you have the ability to control each individual DNA strands. In the origami approach, for example, there are 300 unique strands. You can modify any one of them. You can modify any one of them to recognize any other nanomaterial or any other molecule. So, for example, in this work, the authors created this DNA nanorod. And on this DNA nanorod, because each location has unique, specific DNA strands, you can attach gold nanoparticle in a special way, and in this case, in a spiral. And this turns out to be quite useful because plasmonic resonance of gold nanoparticle can be coupled together. And in this case, because the arrangement is chiral, so the coupling is also chiral. So if you have learned organic chemistry, chiral molecules often have unique properties, especially optical properties, because you can differentiate left-handed right and right-handed right. Now you can do the same thing with nanomaterial, except the interaction with light is much, much more stronger. There are different versions of this. For example, here you have 2 gold nanorods being templated onto 2 DNA rods, and they can be rotated, and depending on the angle of the rotation, you can get different optical response. And even more examples down here. And the common theme in all the studies is the control, either dynamical. So here's a case of dynamical control. What you can do is you can add or remove some strands. And by doing that, you can change the geometry of the DNA structure in real time. So by doing that, you can alter the coupling between the interaction between DNA nanorads, and from there you will get a different optical response. Okay. There are other forms of this. I want to highlight this one. This is a case where different size quantum dots are being anchored onto a triangle-shaped DNA structure. And each quantum dot has DNA strand modified. And you may notice the cartoon here, on the triangle DNA structure, each location has been modified with additional DNA anchor strands. So once you put everything together, you will see this, this is the SEM image. You will see the small dots, those are the quantum dots, and they will align in exactly the way you intended. And this is exciting in a lot of applications because now it offers a way to position different nanomaterial in a very specific way. So you can control energy transfer, you can control charge separation. In many other cases, you have to simply put two things together without knowing how they are positioned in real space. But with DNA nanostructure template, you know exactly, and you can design exactly how they should be positioned with respect to each other. To give another example, I'll stop soon. To give you another example, people also used extended lattice structure to assemble carbon nanotube. This is related to the nanoelectronic application I mentioned a couple of days back. And what they did here is using the DNA break approach to create long channels on a two-dimensional DNA base. And within the channel, there are DNA capture sequence. And at the same time, they functionalize carbon nanotube with short DNA strands or handles. And, and after that, the DNA hybridization would recognize and capture carbon nanotube into those channels. And by doing this, they can create very high density of DNA. And if you look, carbon nanotube array. So if you look at the SEM image here, here is the channel, and here you see with the carbon nanotube in it, you start to see this tubular structure embedded in the channel. I'll skip those and just mention the molecular positioning. Here, the tool is often based on the modification of DNA strands. When you make this DNA nanostructure, you can replace any one of those strands with chemical functionalized version. Okay. So what that means is when you make origami structure, for example, you have the possibility to add a chemistry functional group onto any one of the 300 strands. And you know exactly where those strands would be because you designed those sequence in a computer program. So what you can do is you can precisely position two functional groups at specific location on that 2D platform. And then at a later time, you can attach other things. There are a lot of organic chemistry you can use. You can attach other things onto that DNA sequence. And this R can be another molecule, can be another fluorophore, can be another protein, can even be another nanoparticle. I think I should probably stop here. I'll only mention drug delivery applications where you can use DNA nanostructure as a container. You can modify the surface with functional groups that recognize certain receptor, and that can create targeted interaction with cells. And once everything is into the cell, it can undergo digestion by enzyme in the cell, and that can release whatever drug you're intended to load. there are different formats of this. And another way of targeting drug delivery is you can create channels. You can create a long DA tube, and you can anchor this tube onto the lipid bilayer. And in some cases, you can even add a lock, a blockade at the top of the channel. And you can also add a complementary DNA strand. And by having that complementary strand, you can release this blocking single strand so that the channel is open. Or if you don't have it, then it will be blocked. So I think, apologize, have to skip some of the slides, but hopefully you'll see more of the applications side of things in my next talk. Again, thank you for you taking your time to participate, and be happy to answer any additional questions.
Thank you, Professor Liu, for a very insightful and interesting lecture on DNA nanoparticles and how the scaffolds and strands of DNA can be used for designing some new materials for specific applications like drug delivery, tissue engineering, etc. Now the topic is open for discussion. If you have any questions from the audience, both offline and online, they can discuss. For online participants, they just write down their questions in the chat box or in the WhatsApp group, and somebody will moderate from the organizing team. And offline participants can ask questions directly. I have seen there are a few questions in the WhatsApp group as well. Aditi, can you please ask? Can you please read those questions?
So, how regular structures are made with DNA and how uniformity of shape is maintained? Regular structures are maintained with DNA and how uniformity of the shape is maintained.
So by regular, I presume maybe you're talking about periodic structure. I'm not sure exactly what regular means, but I can certainly comment on the second part. Okay, so let's see. I'll start sharing my screen. Okay, so first of all, on the stability, I have more experience with the stability of individual origami structure. I'm going to show my pointer. Okay, so those individual origami structure, once they are made, they are stable in water, in the buffer, at room temperature, at least for a week, based on our experience. Now, if we freeze them, they can survive a lot longer. Now, there is some damage as a result of freezing, because those are large structures, so the formation of ice crystal sometimes would damage them. Now, once they are deposited on the solid substrate, they maintain their shape for much longer time. In terms of stability, you can view them as the same as any other DNA double helical structure. They have a melting temperature. As long as you don't heat up your temperature above their melting temperature, they will be fine. They are also biological in nature, so if your environment has enzymes that chop up DNA, then that certainly would be bad. So in terms of stability, it would be the same as any other biological, any other DNA material in general. So the other part is how
uniformity of shape is maintained. And for structures, he's asking about regular shape like triangle, trigonal, bipyramidal, etc.
So there are a variety of ways to make them. If you look at this regular structure here is a rectangle, and all you need to do is to fold the DNA in this way. That's all you need to do. If you fold it this way, you'll get a rectangle. If you fold it a little bit longer, you'll get, you can get square or rectangle. And the pattern here actually shows you how the folding happens. So the color, the shade of the color tells you which part the folding started first. So I think from the purple is one end and the yellow is the other end. So in this case, if you look at the square one, the folding actually goes just like this. Okay. If you look at this one, for example, it actually folded in two parts. So it's a rectangle structure, but you fold one side of the rectangle first, and then you go to the other side and fold the other side of the rectangle. So it goes like this, and then do it like this. Okay.
Sir, how we ensure that the functional groups are positioned on right place?
You do not have to, it's probably easier if I use this as an example. So this is a single-tiled approach of making DNA nanostructure, but the idea would be the same in that you have a lot of short DNA strands, but each short DNA strand knows where it needs to be in the final structure. Because if it's in another place, then the hydrogen bonding doesn't work out. So it has only one option, and you know exactly where it's going to be. So when you make the structure, you can chemically modify that strand, and only that strand. And if you do that, you're going to install the chemical functional group only at the position that is supposed to be for that DNA sequence. Did that answer your question? Okay.
Sir, my first question is, sir, what parameters are involved which regulate their shapes? And sir, the other question is, sir, what about other biomolecules which are also available now? RNA biology is taking place. So, sir, have you tried RNA as a molecule in nanotechnology?
I think the first question is what parameters are involved in designing a shape?
Yes, sir.
Okay, so there are different approaches again. Now, if we look at the origami approach, then the consideration is when the fold happens. So that's the only thing you need to think about, when the fold happens. You can just think about a very long wire. You can fold the long wire 300 times in the XY plane. If you have the complete freedom to do that, what can I do to make a triangle shape? There are many different ways I can fold it, but I have to fold it in a way that it goes along the triangle shape overall. So I have to impose restriction that it has to fold in certain way. Now, the good news is there's a computer program to do that, taking into all the helical structure constraint of double-strand DNA. Now, in the case of single-stranded tile, the shape is even easier to handle because you can simply remove some of the DNA strands. The absence of that DNA strand in the final structure would create the shape.
Sir, so for this we use DNA whose whole sequence we know very well. So for that the complementarity of DNA is utilized for this folding.
So in the origami approach would be the complementarity, and we use the complementarity to fold the long strand. In this case, while still everything's underlying principle is DNA hydrogen bonding, but in this case you remove some of the strands. So example would be here, this triangle structure, and versus the whole rectangle structure. If you remove half of the strands in the collection that produce the rectangle, And if all the strands you remove are supposed to be here, then you create a triangle.
Sir, what about other macromolecule like RNA?
I think you asked about.
Sir, like what about RNA?
So the question is, what about RNA? And that's a very good question. So people have used RNA to create RNA nanostructure as well. Same kind of concept. The major difference being RNA is much more chemically liable. So chemical stability of DNA is much worse than that of DNA. So as a result, the research on the RNA nanostructure is not as widespread as that of DNA nanostructure.
Thank you, sir. Thank you, sir.
How I can, okay.
How did you use origami to make? How did you use origami to make DNA origami structure?
I'm not sure what. Oh, so you mean the actual process of making them?
Sir, one slide belongs to DNA origami structure. Then how you use origami, that is mathematics part. How you use origami to make this type of structure?
How? I think you're asking me how do I use origami to make the origami structure?
Yeah.
I'm not sure what. Trying to understand the angle of the. So if you're asking about the actual process of making the structure.
DNA origami, one slide belongs to DNA origami. That is related to that.
Oh, something happened.
Yeah, that is.
This one?
Sir, that is. Previous.
Previous one. This one? Or this one?
So previous. That is.
This one?
Yeah.
So your question is, how do I use origami to make the origami structure?
G. Yes, sir.
Well, I'm not sure what. Okay, I guess the origami would be the analogy the authors are making between folding paper versus folding DNA. The concept here is you fold a DNA long DNA sequence multiple times to create geometric shape. That is just like you fold paper into paper origami. So here you fold DNA into DNA origami. But I'm not quite. I don't think I get your question. If you can rephrase your question, maybe that'll help me.
Okay, sir. Okay. Thank you, sir.
Yes. So we have an online question also. How DNA is protected from WBCs in body for drug delivery before reaching the target?
Yeah. So the question is how the DNA nanostructure are protected from a lot of bad things in your human body before it's able to deliver drugs. That's an excellent question, and this is something. Let me see. Okay, this is something that people actually thought about and started looking into addressing them. As you know, there are a lot of enzymes in your body. So a lot of enzymes are designed to degrade foreign objects and to attack foreign objects. So DNA nanostructure has the advantage of creating this ideal shape. You can create a container, for example, to load the drug, to protect the drug, but also it has the problem because its DNA may trigger a lot of unwanted response, immune response in particular from body. And in addition, it's a biological material, so the degradation of biological material within human body is always a concern. Okay. What has been done, so people recognize this and they have thought about different ways of enhancing the stability of DNA structure. And this one, this is one of such examples where they attached lipid bilayer structure they coat DNA cage with lipid bilayer structure as a way to mask the DNA material from being detected and attacked by enzymes and other proteins in the system. So it is a very a significant concern in my view, and in the future, whether we should use this native DNA as construction material for drug delivery, or we should use them as solely as a mechanism to do fundamental study. I see the possibility of both, but certainly the immune response and the stability needs to be addressed before this can be put in real use. Thank you for the excellent question.
Thank you for very nice lecture. Now we are proceeding for tea. We'll be back here again at 11 a.m. Today we have a short tea break. We'll be assembled again. Thank you.
kindly pay attention. Today we'll give you the feedback forms and you have to fill them.
Varis, where are the feedback forms? Start distribute. Start distributing. Ariba? And how many students have submitted the assignment? 65 students have submitted the assignment. And online? 37 only. So still some of the students have not submitted their assignment. It will affect their grades because they have already passed the deadline. They have missed the deadline. It was 17th and now we are on the last day. We don't have time to evaluate your assignment. If you have to submit it now so that we can have some time during the 2 day sessions to evaluate your assignment. Welcome back to the post-T session. I once again request Professor Hitao to please deliver a talk, and this time he'll be speaking on DNA-based fabrications. DNA-based, the fabrication of DNA-based nanomaterials. Sir, please come upon the dais.
Thank you for coming back. In this talk, I will discuss research from my lab, where we focus on how we can use DNA structure to pattern other materials. So this will be a little bit research focused. I'll try to explain the chemistry aspect of things. If you have any questions, do feel free to interrupt me. Again, I want to thank the people who. Okay. All right. We need to share the slides. Okay, sorry about that. I want to thank the people who did the actual work. A lot of the research was pioneered by a postdoc of mine many years ago, Sumaid Surveyed. He joined my group 2010 and spent about 3 years in my group, developed many of the foundational work that triggered a lot of the later development. Two current graduate students also working on this project, Shubhanka Kundu and also Anamita Kumari. They are here and here. Shubhanka also recorded the video that I'll be sharing with you later in the afternoon, showing the experimental procedure used in our lab to fabricate these DNA structures and their characterization using atomic force microscopy. Okay, so in the morning, earlier talk, I already talked about the power of DNA nanotechnology, and in particular, I want to highlight the high resolution that can be afforded by those DNA nanostructures. We already can use those DNA nanostructures to create materials over multiple length scales from nanometer to almost micron. So I showed the Mona Lisa structure that is approaching micrometer by micrometer length scale. Within the DNA structure, if you think about the double helical structure that's 2 nanometer in size, if you make a turn, that's about 5 nanometer, and I consider that being the ultimate limit of resolution in terms of size. What I didn't emphasize in the earlier talk was the potential of low-cost fabrication, and this may sound counterintuitive because most people would think DNA is a expensive material. Now, I had the same kind of impression that DNA is expensive until I ran some calculations to find out how much does it cost to cover an area of 1 square meter with DNA. Now, the reason I say cover 1 meter area with DNA is in a lot of the experiments I'm going to talk about, we only require 1 monolayer amount of DNA. So only a monolayer amount of material is required, and that translates to very small quantity. So if you run the numbers, you'll find at the current rate cost of manufacturing, it costs about $1 to cover a square meter of area. So within the context of surface patterning, that cost is actually pretty low, and potentially can be even lower in the future. So the DNA template can potentially be a very low-cost template even for large-scale manufacturing. So that's something I want to emphasize. I already showed different ways of making the structure, so I'm going to skip those. A lot of the work, so here's the motivation. What kind of future big direction we're targeting? We're looking at 2 different kinds of future applications. One is in the area of nanoelectronics. And in this case, what we are concerned about is the patterning, doping, and the deposition of materials onto silicon and other semiconductor substrates. So here's a picture of Intel CPU. Now, if you look beneath those packaged structure, what you're going to find is a lot of transistors, essentially, or MOSFET. Now, you're typically shown this kind of electrical engineering diagram, but on the chip, what you see are something like this. You have doped region of silicon, either p-doped or n-doped. You have gate electrodes, and you have gate dielectric, and you have interconnects. Okay. Now, this again is a cartoon. Now, if you look at the actual device, at the chip level, what you see is actually quite different. what you see are horizontal and vertical bar structure. Now, this bar structure actually makes it, those two pictures are equivalent. The bar structure defines the area of doping. It defines where the gate electrodes are, so on and so forth. So what it means is that, you know, if you can make a very simple geometric shape and a pattern on silicon wafer, and if you can make it over a large area, then you can make a lot of transistors. And from there, you can make memory device. From there, you can make logic device. But the first step of making that happen is you have to be able to etch silicon. You have to be able to deposit materials, usually conductor or insulator. And you have to be able to dope silicon. So those are the three fundamental steps of silicon-based nanofabrication. Now, why we're interested in using DNA to make transistors is simply because the current way of making transistor devices become more and more expensive. Now it takes about $5 billion to make a new fabrication facility. This is an earlier number used, quoted by Intel. The extreme UV stepper right now used in a lot of those fabrication cost over $100 million a piece. So this kind of investment would only make sense when you're making large-scale commercial fabrication. You know, a CPU, a GPU, a large-scale memory, you know, things like this. It becomes very expensive to invest and becomes expensive to make. There's another area of patterning we are interested in. That's what I call large-scale surface patterning, and those are completely different incentive. Instead of making smaller and smaller structure, and instead of making regular arrays of those structure, here we're interested in simply creating surface texture. We're not so much interested in making a regular array. As long as we have certain density, we'll be happy. Why this is interesting is because a lot of properties of materials are related to the surface texture. There are certain properties that have nothing to do with material itself. It only has something to do with how the materials are structured in three-dimensional space. Now, wetting is a prime example of this. If you have a porous structure, then when you have water droplet sitting on the surface, the water droplet actually got suspended over by the air pockets embedded between the porous thin film. And when that happens, you will exhibit a superhydrophobic behavior. And that can be beneficial in a lot of applications. Imagine in the cold winter of Alaska, it probably is not going to apply to India. You don't have to worry about ice freezing on your and sticking to your car window. It can simply be brushed off very easily because they do not stick to the surface very well. Okay. There are recent examples where people achieve superhydrophobic behavior using a superhydrophilic material. Okay, this is a silicon oxide material, but patterned into a very strange shape. This so-called double reentrance structure. It's like a mushroom, okay, but it's made in such a way that the water droplet would have to go above to completely wet the surface. So this kind of surface makes the surface super hydrophobic, even though the underlying material is super hydrophilic. So again, the transformation of surface property by using nanoscale texture. There are a lot of interest in so-called metamaterials. There you have metallic or semiconducting materials shaped into very defined patterns. And those patterns can create interesting optical and magnetic properties. And in some cases, can refract light and other magnetic wave in strange directions to the point that you can make certain objects invisible. So you could have cloaking effect by coating the surface with nanoscale and microscale features. So in those applications, we are, as I said, we are more concerned about large-scale patterning instead of high resolution and high density and regular lattice structure. We simply want a lot of those shapes. We want them over a large area, whether they're in an array form or not a big deal. Okay, so this is the second part that our lab has been focusing on. So if I take a step back and think about, the competition between various kinds of patterning techniques, there are more or less two different kinds of patterning. One is bottom up, another is top down. Right? By bottom up, you start with molecular material, and by encoding properties into their molecular structure, they can self-assemble into all kinds of interesting shapes. And in this area, the block copolymer phase separation is probably the most well developed, where you can design the block copolymer structure, sequence, molecular weight, so on and so forth, and they can automatically phase separate into different geometries and compositions. And there are also directed assembly approach developed to, for example, make the polymer phase separation domain into making 90-degree turns. Things like this has been done. Or to incorporate different periodicity into the block copolymer separation. And those are all very powerful techniques. Now, what DNA nanostructure brings, if you look at this diverse shape of patterning templates, it's kind of a combination, offers you a combination of bottom up and top down. With photolithography, you can make any arbitrary shape. With bottom up assembly, you can make regular geometric shape, but not really arbitrary shape. I can't imagine a way to design a block copolymer such that it phase separate into my initial. It's just impossible. But with DNA nanostructure, it's pretty easy to do. If you can make a smiley face, you can probably make anything you want. Right? So with this in mind, we are broadly looking at applying DNA nanostructure as a template for various applications. And I'm going to first focus on nanoelectronics related application, then I'm going to talk about large scale patterning. All right, so the story started about 15 years ago when I started my job at University of Pittsburgh. And I was interested in developing etching process that can be catalyzed by DNA. Okay. And why we're interested in this? Because etching is a fundamental process in nanoelectronics industry. You want to pattern silicon wafer, almost always involve some kind of etching. So we got interested in identifying ways to etch silicon oxide, but we want to do it in the presence of DNA such that we can transcribe the shape of DNA nanostructure into the silicon oxide layer. Okay. And this work was done led by my postdoc, Sumay Surveyed, and I didn't expect it to be working, but turned out to be working beautifully. So what I show here are atomic force microscope images of a DNA triangle that you're probably already familiar with. And those DNA triangles are deposited onto a silicon wafer with a thermal oxide layer. Okay. And the oxide layer is about 300 nanometers. What we did then is we flow hydrofluoric acid vapor over this DNA deposited wafer surface. And what we find was that there are two different outcomes depending on the reaction condition. In one condition, we can create, after the etching process, we can create nanoscale trenches on the surface. And the trenches, the shape of the trenches duplicates the shape of the DNA nanostructure template exactly. And in another case, we find we can create nanoscale ridges on the surface. So a totally different outcome. One is additional etching, another is less etching in the area covered by the DNA nanostructure. So naturally, we were interested in understanding what is happening. And if you think about this, here, the presence of DNA somehow changes the reaction rate and on a very local basis. And you have to think about the geometry here. Here, each DNA structure, the edge length is about 100 nanometer. And the width of this one edge, this triangle, the width of the triangle edge is about 25 nanometers. So this is a very high quality pattern transfer from molecular scale template to the silicon oxide layer. So what is happening? What is happening is a process catalyzed by water. So the etching of silicon oxide by HF is a well-known process and is known to be catalyzed by water. So this reaction will produce silicon fluoride, which is a gas, and water. So this is a classical autocatalytic process, meaning that the reaction would be catalyzed by its own reaction product. So a faster reaction would produce more water, and more water would make the reaction go even faster. Okay. So what this means is the reaction rate would correlate with the amount of water on the surface. Now we're doing everything in gas phase. We have silicon oxide, silicon wafer with DNA on it, and we are flowing HF vapor, and we control the amount of water vapor in the gas phase. So there will be an equilibrium between water vapor and water absorbed on the silicon oxide surface. The presence of DNA would change the amount of water vapor absorbed on the surface, because DNA can enhance water absorption under certain conditions and prohibit water absorption in other conditions. But also what's interesting here is because of autocatalytic nature, even tiny differences in the water concentration on the surface can be amplified by the reaction itself. So with that, essentially what we did here is we can use DNA as a catalyst to enhance oxide etching, but in an indirect way, because the role of the DNA would be a sponge. It absorbs water from the gas phase, and that presence of water would enhance the etching reaction locally. And in each of those trenches, there's a DNA structure sunken into it. Okay. Now, if we change the relative humidity to a different regime, you will run the reaction at a higher temperature. It turns out the DNA is not very effective holding the last bit of water. In that case, there's a competing process where DNA becomes a diffusion barrier, and that makes the overall reaction rate lower. So we can switch between these two by simply changing the reaction temperature, which changes the relative humidity of the environment. So with this idea being figured out, we look at a range of questions, and in particular, what is the ultimate resolution we can get? And the DNA structure turns out to be particular convenient for us to answer that question, because the structure we use actually has a single-stranded tail of DNA. This is about, I think, 70 to 100 bases long. It just hangs there, and there's no double-strand structure. That's by design. And what we find is in at least some of the etched structures, we find this small tail in the transferred pattern, and that indicating that even a single-stranded DNA is capable of delivering this catalytic effect to the substrate, and you can essentially carve the shape of a single-strand DNA into a silicon oxide substrate. We are able to get around 10 nanometer, 15 nanometer lateral resolution, and we can make pretty deep trenches up to 11, and later we can push this to even 20 to 30 nanometers deep. And the etching selectivity is at least 3:1, so the presence of DNA would enhance the etching rate by a factor of 3. More recently, we started to think about ways to incorporate or modulate the etching rate. And the thought process here is that because everything is controlled by water, So what can you do to change the amount of water on the surface? With a background of DNA plate, you can add additional material, a protein in particular, because a lot of protein, their external surfaces are hydrophilic. So we are able to show that by modifying DNA nanostructure with streptavidin, this is just a convenient protein for us to use, we can make the etching a little bit faster in the area being modified with streptavidin. And by doing this, we can create a pseudo 3D fabrication instead of just doing a 2D fabrication. And to characterize this, we end up have to use a polymer replication process because the trenches are actually too deep for the FM tip to probe the bottom. Some of the images I show are actually the replicated polymer substrate. And this shows you the general picture here, and I want to highlight this one. This is the triangle-shaped DNA nanostructure, but with one side of it modified with proteins. And you see on this one, this is the contrast enhanced. You see one edge is more blackish in the red background. Without contrast enhancement, you don't really see it in the regular color scale here. But once you change to a different color scale, you find a lot of them are one side modified. And in the histogram, you see one edge is higher than the other two. So with this modified DNA nanostructure, we are able to conduct the etching, and this is the polymer replica. Again, here you see one of the edge, this one being higher than the other two. And if you look at the histogram, in this case, we're etching as deep as 25 to 30 nanometers. With the modified structure, we see about 10 nanometer enhancement of etching depth. So on the edge that has not been modified is about 25 nanometer. After modification, it's about 35. This one shows a high resolution image on the polymer replication. And again, a deeper trench becomes a higher ridge because it's inversed. Here you see one side is higher than the other two. All right, so another process we've been interested in is to think about how to make a conductive nanostructure. You recall the overall scheme we have to accomplish. One is etching, another is some kind of deposition, and the deposition oftentimes involve conductive substrate because you have to create conductive wires to wire up different nanoscale electronics. So at one point we got interested in figuring out whether we can convert DNA nanostructure into a conductive carbon material. And this may or may not ultimately solve the need for nanofabrication of electronic devices. But I thought it's an interesting example of how an organic nanostructure can be used as a precursor to make inorganic materials. So what we demonstrate here is that DNA nanostructure being organic, you can view them as feedstock. And if you coat the surface with a thin layer of oxide by atomic layer deposition, we can trap the organic material in the matrix. And once we do that, we can heat everything to high temperature and carbonize all the organic material. And that turns the DNA into carbon material. And those carbon materials are conductive, as we shown by conductive atomic force microscopy. So this shows the DNA nanowire template we used. And then we coat the nanowire with atomic layer deposition. Now the coating is pretty thin, so everything is conformally coated. So if you look at the atomic force microscopy image, you don't really see anything different. You still see the same kind of nanoscale feature of DNA nanowire. What you do not know is there's a 20 nanometer thickness of coating on it. We're able to show that after annealing, if you look at these two images, these two are taken at the same time. This white square, okay. After the annealing, we're took the AFM image at exactly the same location, and you find the patterns are still the same. So the annealing, the high temperature annealing doesn't damage anything. And we also removed the silicon aluminum oxide coating to expose the underlying carbon nanomaterial. Again, the two images are taken at exactly the same location. And I want to mention this is very, very difficult to do. Took my students quite an effort to relocate exactly the same location on this. If you think about the silicon wafer we use is several centimeters in size, and somehow you have to find the same area within a micrometer, 100 nanometer precision. Okay. But the message you can see here is that those two patterns are exactly the same. Okay. So whatever was DNA is now being converted to amorphous carbon material, and we did conductive FM and showing that those materials are conductive. And more recently, we started to think about the doping, because doping is a fundamentally important role and component in semiconductor fabrication. And we're thinking how we can dope silicon using DNA, and how we can change the doping concentration by manipulating the molecular chemistry of DNA. So related to this, many years ago, we studied how the DNA nanostructure would decompose under very harsh conditions. And there we find a very unexpected conclusion that once you deposit DNA structure onto silicon, there are actually the shape of that is actually very robust. So as an example, here we have those triangles on silicon wafer, and we subject to UV ozone oxidation. This is a very harsh oxidation condition that pretty much guarantees removal of all organic materials. And that's our expectation as well. However, if you look at the AFM image after 20 minutes of exposure to UV ozone, you find the surface is still full of those triangle features. Okay. Now, we know that the 20 minutes exposure actually got rid of all the organics. And the triangle shape, if you look at the cross-section, actually has a much reduced height. So we are actually indeed removing the organics. What is left is the inorganic material. And what is amazing is that those inorganic residue maintained the overall shape of the DNA structure. This is, in retrospect, is not very unexpected, because on the solid surface, the diffusion is going to be very slow if there's no water or liquid to facilitate the diffusion. So the phosphate residue, because the DNA has phosphate backbone, and that phosphate backbone is not going to vaporize in a typical oxidation condition. So the phosphate would stay behind in the form of phosphate, metal phosphate, and that phosphate would stay behind and in the same shape of the DNA structure. So with this in mind, we start to think about ways we can leverage the phosphate residue for doping. And the reason being, phosphorus is one of the most common dopant used by silicon industry. So phosphorus has 5 valence electrons, silicon has 4. So you can dope phosphorus into silicon, that will make it into N-type doping. Okay. So that's what we did here. We deposit DNA structure onto silicon. Now this time there's no oxide layer. We get rid of the DNA, organic portion of the DNA, and we were left with residues. We coat it with a layer of silicon oxide, and the intention here is to limit the lateral diffusion of the phosphates. And then we heat it to high temperature using a process called rapid thermal annealing, RTA. And what happens in this here in RTA is under intense temperature, about 1,000 degrees Celsius, there will be a solid state reaction between phosphate and silicon, and that reaction will produce phosphorus atom, which then diffuse into silicon to dope the silicon wafer. What's unique in this process is that the doping would only happen where there's a DNA nanostructure coverage. It will not happen when there's no DNA. Okay. it also, the doping density is also a function of the amount of DNA base pair, because one base pair translates to 2 phosphates. So if you have a single layer DNA nanostructure, it will give you lower doping density compared to the case where you have double layer or tri-layer DNA nanostructure. So this gives us a way to modulate the doping density in one batch of doping process. Okay. Now, in the traditional nanofabrication process, each doping step will only give you one doping concentration. Okay. We also tried an alternative method where we use sulfosym monolayer to inverse the polarity of the dopant. So with DNA, you can do N-doping because there's phosphorus, but that's the only dopant you have in DNA. To achieve P-dopant, usually you would need something like boron. Okay, but there's no boron in DNA material. So what do? Instead of doing it directly, we deposit DNA onto silicon wafer. We grow a sulfosym monolayer. Now in this case, the DNA would block sulfosym monolayer happening everywhere. And we get rid of the DNA and then refill the gap using a boron-containing sulfosym monolayer precursor. So now we can create a patterned sulfosym monolayer where there's a combination, there's a boron-containing molecule at where the DNA nanostructure is being deposited. And we can do the same rapid thermal annealing, and by doing that, we can create a doped area where the doping is boron in nature instead of phosphorus. Okay, so with that in mind, I want to show you some actual data. So here we used a wire-shaped DNA nanostructure. It's easier for us to demonstrate the case. So this is before, this is after the DNA nanowires are deposited on the surface, and then we expose it to UV ozone, and what you see is the height of the structure become much less, and that's because you're oxidizing the organic portion of the DNA. Now, after the doping, what we find are two changes in the AFM. When we look at the topography, we start to see a lot of trenches on the surface. And if you recall the message here, there's some kind of solid state reaction that etches silicon. Now, if we look at the surface potential using Kelvin probe microscopy technique, we find there's also contrast in the surface potential. And this is one of the probably the strongest evidence indicating that the silicon is being doped. So the contrast here is not due to topography, but rather a change of surface potential, which is a function of dopant type and dopant concentration. Okay. So with the doping process figured out, we collaborated with a research group in electrical engineering, and we evaluated the doping process in the context of fabricating nanoelectronic devices. And here we made some very simple devices, a p-n junction, we have a diode, and what we did is we simply deposit one contact on top, and we use the silicon the silicon as the back contact. And in this case, we start with p-doped silicon, and we dope with excess amount of phosphorus. So this is an inversion of the doping profile, and that created the p-n junction in this vertical direction. And we can make a diode structure, and by measuring the IV curve, we find this fits our typical idea of p-n junction. The conduction is one direction, but not in the other direction. A control device showed no particular non-doped device showed no leakage current based on our fabrication technique. Okay. We also fabricated more complicated structure, three-terminal field effect transistor device. Here we start with a DNA nanowire, and we dope an intrinsic silicon wafer. So in this case, there's no doping to begin with. Again, we dope it with N-type dopant phosphorus. And we use conventional lithography technique to define a field effect device in the middle. And this is the actual device with wire bonding. And if we measure the conductance of this channel, we find the conductance goes up significantly. That's the red. Up to remind people that this is voltage as a function of current. It's the other way around. So a flat line indicates high conductance. Okay. And a sloped line indicates low conductance because this is the voltage drop after applying this amount of current. So it's the opposite of RV curve. This is a VI curve. Okay. But this shows that after doping, the electrical conductance got greatly enhanced, something you would expect. We also conducted gating measurements. So this is a transfer curve where we look at source drain current as a function of gate voltage. And what you see here is the modulation of source drain current as a function of gate voltage. And that, again, is what you expect for a functioning transistor device. Now, if you look at the data, you'll realize that we're actually cheating quite a lot. The bottom is not zero. We're not turning off the transistor. Okay. We're actually, if you look at the actual modulation, it's actually pretty small. It's from 1.8 to 1.9. We're looking at 5% modulation, which is not a great device. And we're not claiming it's a great device. What is exciting in this device is that we show using DNA, you can dope a silicon wafer with nanoscale confinement based on the shape of the DNA structure. And from there, you can actually make, not meaningful, but you can make proof of demonstration device. And certainly, if you look at this one, this is a much better device characteristic curve based on typical expectations from electrical engineers. So there's still a long way to go. Now we have ways to do etching, we have ways to do doping, and the rest of the question is how can you make instead of one. So here, all this wire bonding is for one device. This is fine for research purpose because we want to figure out whether we can make one device, and the answer is yes, we can make one. Now the next step is, can you make 10,000? And that's something we want to accomplish in the near future. And if we can make 10,000, that means we can probably make a logic unit of a computational unit. So the smallest CPU, the 4-bit CPU, would require about 4,000 transistors. Now, if you think about the Mona Lisa figure, that's about 800 by 800 nanometers in size. Now, if you also think about what is the smallest feature you can create within that big chunk of space, I would say about 10 nanometer by 10 nanometer easily. Right? So if you do the math, you'll see that within that Mona Lisa, figure, if we change the pattern instead of making Mona Lisa, what if we change Mona Lisa into a mask of a 4-bit CPU? How many transistors we can make? We can potentially make 80 by 80 devices, and that's 6,000 devices using one mask of DNA template. If we can do that, I think that will transform the way people think about nanofabrication, especially nanofabrication using bottom-up approach. All right, so with the rest of time, I want to discuss a totally different direction of nanofabrication. And this would be a case for large-scale application. And I would call them probably cheap application. Here we are way more interested in cost, how we can reduce cost, how can we make it really cheap so that people will actually use them. The story started with the same kind of DNA template oxide etching that I showed you before. Except the interest here is not making a highly precision shape, but rather all we want is to create nanoscale roughness. And to some extent, we also care about exactly what kind of nanoscale roughness needs to be created. But at higher approximation, as long as we can create nanoscale roughness and we can create the dimension of those roughness, we'll be happy. And with DNA template, we can certainly do that. So here I show different silicon wafers, not subject to any HF etching and subject to HF etching, but without applying any DNA templates, and also subject to HF etching using different surface coverage of DNA templates. What you see here are certainly different degree of surface nanoscale roughness. What we find is that a rough surface at the nanoscale produce less adhesion of bacteria. This is, again, nothing new. People know this already. And the data is shown here. And what you see here are the silicon wafers, those 4 different samples. We incubate them with different kinds of bacteria suspension. After a couple of hours or after a couple of days, depending on the experiment, we take it out. And we basically look at how many bacteria are being absorbed onto the surface. Okay. And if you look at the data, so those are all stained. So each dot is a bacteria, essentially. And in some cases, they aggregate. In other cases, they don't. Okay. And there are 2 different channels. One bacteria. So if you stain everything with PI, this is one dye that only stains dead bacteria. And Cyto9 stains both live and dead bacteria. So what you see is the great reduction. You look at this too. The very significant reduction of bacterial adhesion to the surface. And that is entirely due to the nanoscale roughness. Because bacteria has a dimension of a couple of micrometer. And when they come to the surface, they have to have effective interaction surface area for the bacteria to stick to the surface. And once you make the surface nanoscale rough, you reduce the effective contact area, and that translates to reduced bacterial adhesion. And this reduced bacterial adhesion can be beneficial in a lot of contexts, in particular medical device, implants, catheter, for example. You want to reduce bacterial adhesion on the surface so that infections and a lot of other undesirable medical complications can be reduced. This again is a long-term study showing that not only the initial bacterial adhesion can be reduced, but the biofilm growth, this will be the long-term outcome of bacterial adhesion. You grow bacterial colony on the surface, and we were able to show that a high density of the DNA nanostructure can produce the kind of nanoscale texture that reduce also the biofilm formation. Okay. So that is one direction in the context of biological material or medical device. Now we are looking into even cheaper ways to expand the fabrication into other materials. Silicon oxide is good for proof of demonstration, but you're probably never going to find a medical device made of silica. So what we're doing now is to reproduce everything we demonstrate here on a polymer substrate to show that polymer substrate can also be patterned by DNA nanostructure, and their nanoscale morphology also translates to anti-fouling behavior. Okay. We are also looking into ways of applying this surface texture trick into modulating optical properties of surfaces, and in particular reflection. This has a lot of relevance in everyday consumer electronics. If you pull out your phone, and a lot of times you'll see this glaring if you are viewing your phone or your TV under bright sunlight. and you're probably very annoyed by the strong reflection. And how can you reduce that reflection? One way to do that is to, again, create surface texture. So reflection happens when you have a sudden change of refractive index. However, if you create a gradual change of reflective index, then reflection will not happen. And that's the foundation of many anti-reflective materials. And here we show that we can use the nanostructure. Those are, again, those triangle structures. We don't really care what kind of structure we use for those experiments. We only want to show a proof of demonstration. And it just happened that this triangle structure has been very convenient for us for a lot of applications. So we were able to show that we can, in addition to doing the etching, we can also do additive manufacturing, depositing all kinds of oxide material onto the DNA nanostructure. And from there, we can conduct conventional plasma-based etching, and that produce high aspect ratio structures. And you'll find a lot of those triangle, there are columns now. You're looking in the scanning electron microscope, looking down. And looking at the optical performance, the nanostructures that has been patterned by the DNA, the blue curve, shows much reduced reflectance by about 40 to 60%. Okay. And the control samples is at 100%. And if you just deposit DNA mask but not doing the etching, pretty much no change. All right, so I just mentioned that we're interested in patenting polymer, and with a DNA nanostructure, we tried a different approach. Here we demonstrated that you can use DNA nanostructure as a stamp, as a physical stamp, to imprint its shape by mechanical force, to imprint its shape onto polymer. So on the top, again, are those triangle structures that we used. And on the bottom are polymethyl methacrylate polymer surface. And those polymer surfaces show the negative imprint of the triangle. And those are done by spin coating the polymer onto the silicon wafer with triangle deposit on silicon wafer. We then peel it off, and somehow the shape of the DNA got transferred to the polymer side. And you'll find in many cases the quality of pattern transfer is very high. The DNA nanostructure here are actually made of 3 edges, and there's a small gap between each of the edges, and you can see somehow, you can see that small gap being reflected on the inverse replica. So with that in mind, we started to think about more direct imprinting techniques. So one direction we're going right now is to study how DNA nanostructures can be directly deposited onto polymer. And we have some initial success, but not quite a lot. DNA nanostructure is very hydrophilic, and most of the polymers are hydrophobic. So traditionally, people didn't think about those two are compatible. But our recent work, still unpublished, seems to suggest some kind of hydrophobic interaction and incorporating charge-charge interaction can stabilize the DNA structure even if you are working with a hydrophobic polymer substrate. So with this kind of process in place, we may be able to work with DNA nanostructure templates on a wider range of polymer substrates and develop etching and deposition reactions directly on those polymer substrates. So with that in mind, I think I'll stop. To summarize, I hope I convince you that DNA templates can not only serve as a high precision fabrication process, but can also work very well in areas where low cost is also important. We have used DNA nanostructure in a variety of template substrates. We spent a lot of work on silicon oxide, but the more recent work suggests that even organic, hydrophobic organic templates, substrates are also compatible with DNA nanostructure. Again, there's a lot of counterintuitive results there. But hopefully you'll see DNA now as a more, it's more than a genetic material. We still use a lot of this genetic encoding technique to control the shape, to control the properties modifications, but you can also view it as a structural material. Especially in the nanoscale, it can be a particular interesting structural material to define the chemical reaction and to encode geometric information. So with that, I thank you again for being here, and I'd be happy to answer any questions.
To deposit nanoparticles on a silicon wafer, as Professor Hetao has explained that how we can use DNA as a platform. First of all, we can introduce nanoparticles on a DNA and then put that DNA on the surface of silicon wafer, and then we remove the DNA by either UV or ozone oxidation, so that only the nanoparticle would come on the surface of the silicon wafer. So it can be used as a platform. You may ask questions, anything related to the fabrication, how this can be done, what is the cost maybe, what is the cost of, Professor? How economical is it to use DNA as a...
Yeah, that's a very good question. So when I started my independent career, I thought, oh my God, this is going to bankrupt me in a couple of years. So we ordered a batch of, those triangle structure, those are made by the origami approach. We bought one batch of those 300 DNA strands from IDT. I don't know if they have a branch in India, but it's a popular vendor for custom-made DNA strands. So we spent about $3,000 on those 300 strands. We're still using them today, and that was ordered 15 years ago. So it's very economical. Yeah, we more than, actually, if you do the numbers, the scaffold strand is actually way more expensive. But because a lot of the work we do is about monolayer amount of material, and each experiment only consumes wafer chips, just a small wafer chip about 2 centimeter by 2 centimeter. The amount of material we consume is actually minimal.
Give her the mic. Give her the mic.
If we have more complex DNA based material, then how can sequence specific interaction can be optimized? If we have more complex DNA based material, then how can optimize sequence specific interaction?
So the question is, if we have more complex DNA material, how can we optimize sequence-specific interaction?
Interaction, yes, sir.
Okay. Thank you. So the sequence parameter space is always going to be limited by the available, I guess, well, the more DNA material you have in one structure, the more likely you're going to encounter certain sequences. For example, in the context of making the origami structure, you have 300 unique sequences, and that certainly would limit on what new sequence you can use, because you want to avoid, duplicating the same sequence that is already there. That can be a problem, but usually, even for the typical origami structure, we're only talking about 300 unique strands. If you think about each strand is about 20 to 40 bases long, the parameter space is actually huge. We're talking about 2 to the 20 or 2 to the 30. So in that case, it's still not limited by the parameter space. It's not a real problem yet. Of course, if you are going to create a larger structure, it will become a problem. A benefit of the structure is a lot of the sequences are being protected, so to speak, because they are double helical structure. And if there's no niche, then strand displacement in a lot of cases are pretty slow. So even if you have conflicting strand sequence, sometimes it may or may not really impact your work.
Sir, what are the challenges in scaling this technique?
Can you repeat?
Sir, what are the challenges in scaling this technique?
The scaling. So there are a number of, areas. One is the DNA material itself. So there needs to be a large production of DNA nanostructure available so that people can actually use them. There are a couple of companies already selling different kind of DNA nanostructure. Now, if the application is 2D related, then I think in most cases the cost is not a real big issue. However, DNA is still expensive if you are calculating things on a mass basis, by a per gram basis. DNA is still quite expensive, and that is in the context of our, machine-based DNA synthesis. There are ways to go around it. For example, you can do fermentation. You can use biotechnology to produce libraries of DNA strands, and from there you can, make DNA structure. And that has been done. So you can grow bacteria essentially, and the bacteria will spit out DNA structure. So there are ways to mass produce those structure, but I have not seen a killer application yet. Those are all new technology, a lot of exciting things that can be done. But is there a commercial product that has to use DNA structure? I see a lot of justification in the lab, in fundamental research. It can provide a lot of benefit. But in the industrial setting, I have yet to see a strong justification. So we still are looking for a good application that justify the use of DNA nanostructure. What kind of so? So the question is potential application of DNA nanotechnology in packaging application. Do you mind elaborate what kind of packaging you're looking, or what kind of potential use of DNA nanostructure in that context? Right. So antibacterial packaging. So in that context, I think, again, my view might be biased. I see some role of creating surface nanoscale texture to make the material intrinsically bactericidal. So for example, if you create a surface where there are a lot of sharp needle-like structure, can you make the surface, in that case, killing bacteria on contact, because there'll be surface tension force that can apply to bacteria so that the needle-like structure could puncture a bacteria on contact. Now, that kind of concept has been demonstrated in some surfaces, like silicon in particular. Now, can you apply that to another material that people use in packaging? probably and maybe, but I do not know exactly how that can be done. If you are thinking more along the line of chemical-based antibacterial, then antibacterial surface, then I don't see a strong justification of DNA nanotechnology there. Conventional organic chemistry, surface coupling, things like this might be more useful or straightforward.
One more question.
So what are the applications of DNA origami? Applications of DNA origami.
You are asking the application of DNA origami.
Yes, sir.
Okay. I think I elaborated a little bit more on this side in the previous talk, but briefly, I would say the uniqueness of this class of material is the ability to define its shape and define material composition and the position. So whenever you have an application that requires a precision placement of molecules or materials, DNA structure become relevant. So in that context, any lithography, any patterning would make sense. Any energy transfer related application or cascade enzyme catalysis would become relevant. What else? Sensing, and especially in the context biological system, because sometimes you want the precision placement of different receptors or the same receptor but in a particular array form, and sometimes this kind of arrangement can trigger biological reaction and produce downstream cascade biological signaling. So those are the broad range of applications I think would justify a strong justification of DNA nanotechnology involvement.
Sir, what are the limitations of DNA-based nanoelectronics?
So the question is about application of DNA-based...
No, sir, limitations. Limitations of DNA-based.
Limitations, okay. So limitations of DNA-based nanoelectronics, I would assume you're asking for DNA-based fabrication of nanoelectronics. That's a very good question. So I didn't get a chance to talk about the downside, but if you, well, I think I mentioned some of it, but if you look at the example I showed here, we're talking about one device. Right? Now, if you look at the motivation slide I showed, this CPU has probably 1 billion of devices, probably even more. Okay. Now, and in this case, the devices are in array form, so you have an array of transistors, especially when you have a memory, you have an array of capacitor, you have an array of 2-terminal, 3-terminal devices, and you have to do this over 1 billion times, and every time it just works. Okay. Otherwise, it's a disaster. Now, semiconductor industry is able to do this after 50, 60, 70 years of refinement, and the DNA technology is not there. So the first order challenge I see it is a large area fabrication of device array. How can you make an array of device, and even before that, how can you deposit DNA nanostructure template into a regular array with minimal defect? There's no good answer and no good solution to that. There's some people already work on this and they have very creative solutions, but from an engineering perspective, the solution is not there. Okay. In addition to that, a lot of those devices, they require multi-step fabrication. With the DNA template, we're not there yet. We're able to do one-step fabrication, and the DNA nano templates are gone. Now, if we're going to do another step of fabrication, we have to make sure those two steps are aligned. That, again, is a big challenge. how can you deposit the DNA structure to the same location, etching, another step of doping, and then third step of deposition, and somehow everything just aligns with nanometer precision. Again, there's no good solution. So there's a lot of challenges, and maybe that might be a shortcoming and issues that I didn't get a chance to elaborate.
Sir, how significant is the type of DNA and the source from which it is obtained?
Can you repeat?
How significant is the type of DNA and the source from which it is obtained?
You are asking the type of DNA
and the source from which it is obtained.
So all the approaches that people use here, in this case the bacteriophage DNA is the long strand, so that's biological in nature, and all the short strands, they are synthetic, made by automated DNA synthesizer. Okay. Now you can certainly, I mean, DNA is DNA. That doesn't matter which source it's coming from, it's still the same. So as long as the sequence is the same, doesn't matter how you make them. And there are ways to make all the components from bacteria fermentation process by regular standard chemical biology approaches. That's not a problem. The future and sorry, the last part I didn't catch. You're asking the future and direction of DNA nanotechnology. That's a very big question. Here's my very limited perspective. I see two strong justifications. One is very fundamental research where cost is not an issue. In that case, you can justify using very expensive templates to create the kind of distance information you need to advance basic science understanding. Okay. That can involve, cascade catalysis using enzyme and other things. That can include precision distance-based measurement of energy transfer, so that you can obtain very useful fundamental understanding of your material system. Drug delivery can have some future, I think, but it needs to be the imaging and drug delivery. The biggest hurdle I see is the stability and biocompatibility. But with that in mind, DNA structure can be converted into many other things. So you could just be using DNA structure as the first step, and then produce some other carriers that better suits your need. Other than that, I see the high resolution patterning may have niche applications, because this can be a way to, if you combine top-down and bottom-up, this can be a way to pattern high-resolution features at much lower cost. And certainly my own hope is that the low-cost application can materialize in the future. We've been pushing really hard in those directions, but because I'm working on that, maybe my view on that is also biased.
Anyone else? Want to ask question? Anyone else?
So my question is, in fabrication, what kind of interaction exists between inorganic phosphates of the DNA and the silicon dioxide? So,
very good question. So the question is, what is the nature of the interaction between the phosphate and the silicon oxide so there are a range of different context. So first of all, when the DNA structures are deposited from solution to the surface, there is ionic interaction between the phosphate and the silicon oxide surface mediated by magnesium. So phosphate is negatively charged, but silicon oxide solenoid surface, when it ionizes, it will produce silicon oxygen minus. So the surface of silicon oxide is also negatively charged. But somehow these two negative charge species do come together. And the reason being, when you do the deposition, there's a high concentration of magnesium 2 plus ion in the solution. So the magnesium 2 plus serve as an ion bridge. So silicon, oxygen, magnesium, and then phosphate on DNA. Okay. So that interaction turns out to be important in controlling the degree of surface coverage. And I think it also helps to maintain this morphological stability of DNA structure. Now, after the deposition, once everything's dried, then there's no mobility per se. And if you remove all the organics, I would assume that it's just, inorganic small clusters of magnesium phosphate, deposited on silicon oxide. Thank you.
For a very insightful lecture. Please give a huge round of applause for Professor Hitao.
We're down here in the morning.
Okay. Start your tutorial after that.
Yes.
And have some rest.
Yeah. So I'll.
If you want, you can remove the
So
camera.
yeah. So I'll have. I need to copy everything. I have a very large video. I have to copy to the computer.
Okay.
So I have a 15 minutes video,
And then
and I have a couple of slides explaining what students are doing.
You can do so.
Yeah.
During the lunch hours. Today, because I have another presentation.
Okay.
I'll give that presentation.
Okay.
Me and Dr. Asfar.
Okay.
And during the lunchtime, we'll transfer all the stuff.
Yeah. Okay. Do you want to do the tutorial first, or do you want me start the video
After the
first?
lunch will be better, I think.
You want me to start first?
No, I think I should complete it.
Okay.
And your tutorial and video after lunch.
Wait, you're doing right now?
Yeah.
Oh, okay. All right.
okay.
I'm interested.
You
Yeah.
may rest.
Yeah.
Okay.
Okay. I thought
We can start lunch at 1. In the post-lunch session, Professor Hitao will show you the video of his own lab, how they have deposited DNA on silicon vapors and how things have been done in this particular direction. So he'll show you a video of his own lab. The work has been conducted by his student that will show during the tutorial session post-lunch. Thank you. We'll be starting a PPT, short PPT on the instrumentation technique that you have been demonstrated yesterday on Raman and XRD. During the lunchtime, you are also advised to complete the feedback form and the test, MCQ multiple choice based test, will be held immediately after the lunch, followed by which we will be having the tutorial session, the last tutorial session and the last session of GIAN program by Professor Hitao at 3 o'clock, and 2 to 3 we will be having MCQ test. Good afternoon. Yesterday we had visits to the Interdisciplinary Nanotechnology Center as well as the University Sophisticated Instrumentation Facility. And we have been very fortunate that the team of USIF and INC, they have explained the instrumentation. They have arranged our visit followed by the demonstration of some advanced scientific instruments, especially the XRD, X-ray diffractometry, the Raman spectroscopy, scanning electron microscopy and transmission electron microscopy. And yes, of course, XRD single crystal was also there. I thank the whole team of USIF and INC. It was very useful and enlightening experience for the students of the GIAN program. Although most of the time scholars are a bit aware of these techniques, research scholars are using these techniques, but most of the times PG and especially the UG students have not even seen those advanced equipments. So it was a very good opportunity for the students to see the machines, to understand the working principle and how the sample is being studied, the experimentation has been done. This has been explained in a very detailed manner. I have collected feedback from the students and even I was also present physically in both the centers, INC and USIF, and everything went very well. The participants enjoyed a lot, they learnt a lot. Although it was planned that before moving on to those centers, we'll give you a brief information about those techniques, but due to our engagements, we could not give you the presentation through these PowerPoint slides. I just give you a brief idea yesterday without using the PPT. So today since we have time also, I have been explaining you these two techniques. The Raman spectroscopy. The Raman spectroscopy, Raman spectroscopy, as I said, it is a complementary technique which completes the IR technique, especially those molecules which do not have a permanent dipole moment and which are IR inactive. They can be studied via Raman spectroscopy. The best thing about this technique is that it has been developed by our own scientist, Sir C.V. Ramanji, and he discovered this phenomena of Raman scattering on February 28, 1928, for which he got Nobel Prize in 1930. And since then, the government of India has decided to celebrate that day when Sir C.V. Raman has discovered this phenomena, and therefore we celebrate it as National Science Day on every 20th February. All the research institutions, universities, colleges, various departments of AMU, we celebrate this day by having some lectures, some talk on Sir C.V. Raman, his research, his life, and how important is for us to be more of scientific temper, how important it is to nurture scientific temper in our student. As I said, it complements IR, it is quite related to IR, so it is also based on vibrational as well as rotational motions. Just like we have UV, which is an electronic transition spectroscopy, also known as electronic spectroscopy, and since electronic transition requires the highest amount of energy, so we need energy of UV region, which ranges from 150 to 190, it is called as the vacuum ultraviolet region, then from 190 to 360 or 380 UV region, then from 380 to 760 we have visible region. But for vibrational and rotational transitions, the energy required is not very high, So we can have it with IR radiations, which have a little bit less lower energy when compared to UV and visible. The principle of Raman spectroscopy. When light falls on matter, when light interact with a sample, this interaction either it may be elastic or it may be inelastic. When the interaction is elastic, nothing will change. The energy wavelength of the light that falls on the sample, because it is an elastic interaction, the same amount of energy is being carried by the scattered light. Since there has been no change, it is of no use. While in case of Raman scattering, that in elastic scattering is called as Rayleigh scattering. Now we have some inelastic scattering as well, which is called as Raman scattering. And whenever this inelastic scattering occurs, there are again two possibilities. In one of the possibility, on the other hand, there is another possibility that the scattered light has more energy. This condition may occur whenever the molecule is in excited state. It comes back, it releases some energy along with the energy of incident radiation. So when it gets energy of the deactivation of the molecule as well, so that energy becomes even more than the energy of the incident radiation. In the former case, we have Stokes shift or Stokes lines, while in the latter case we have anti-Stokes lines. Anti-Stokes lines. So as this slide is showing, in case of Rayleigh scattering, the photons of the light which is falling on the sample, they do not get any energy, they do not even lose any energy, because it is a type of elastic collision. All the energy is conserved. However, on the other hand, when it is an elastic interaction, in inelastic collision, in elastic scattering, the incident photon may lose some energy or gain some energy. When it loses energy, the energy of the scattered line is lower. It is called as Stokes lines. On the other hand, we call it as anti-Stokes line. And in any of the spectroscopic technique, or I should say in all the spectroscopic technique, there are certain few components which are integral part of that machine, which must play some important role, without which it is not possible to complete the study. Out of those components, the two major components are the light source and what is the next? The detector. Although we have monochromator in all those spectroscopic technique, and a monochromator consists of, what does monochromator consist of? Slits and something to break light. It may either be a grating or a prism. So for a monochromator, we need either a grating or a prism which breaks the light into individual colors, and then we have entrance and exit slit to select a particular wavelength of light while deselecting the others. So that monochromator is of course used, but the main components are the light source and the detector. In case of Raman, we use laser light as a source, the light amplification for stimulated emission of radiations, while the detector which is used is the charge coupled devices which collects the photons falling on it and convert the intensity of the photons into an electrical signal. So Stokes lines or Stokes scattering means the scattered photon has somewhat lesser energy than the incident photon, and it will happen when the photon of the incident light transfers its energy to the molecule, resulting into the excitation of the molecule. When molecules undergo excitation, it absorbs energy from the incident radiation, resulting into the loss of energy of the scattered photon. While on the other hand, in case of anti-Stokes scattering, the molecule may already be in excited state. The molecule deactivates, releases energy. That energy is being taken up by the photons, and the scattered photons have higher amount of energy than the incident photons. Energy. While in case of anti-Stokes line, it is the opposite. When it is released, when it is scattered, it has more energy than the incident photon. The same has been explained over here. I think if you remember Professor Hatau's lecture on day 1, while he was talking of graphenes, he showed you various spectra of Raman, and there he shows that Raman spectroscopy is one of the best technique to characterize graphenes. It helps you to study the disorderness or the defect density of graphenes. Just by measuring the height of D and G bands of Raman spectrum, we can identify the defect density or the disorderness of graphenes. So by looking on the Raman spectrum, you may identify whether your graphene sheets have defects or not, and if their defects are there, what is the density of those defects. So it is one of the very good technique for graphenes. So there is a term called as Raman shift. The Raman shift is basically calculated by just measuring the frequency difference between the incident light and the scattered light. That frequency difference is basically called as the Raman shift. That frequency can be converted into energy by E is equal to h nu equation. This Raman shift is given in the form of wave numbers, just like we have in IR. In IR also you must have seen the IR spectra, where we have intensity on y-axis and on x-axis we have wave numbers. Can anybody tell me the light source of IR? Which light source is used just to make it more interactive? Anyone? Any guesses? Other than teachers? Is tungsten? No. It's a nurse glower which has oxides of certain metals. When it is heated at high temperature, it starts emit IR radiations. And do you know that IR radiations are very useful in night view cameras as well, because IR radiations have thermal energy as well, they carry thermal energy, and those night vision cameras are nothing but they are based on IR radiation. A body which is hot is being shown red or orange color, while colder bodies are shown in light colors, blue color. Have you heard about a telescope called as JWST? James Webb Telescope. I must have sometimes explained it in class. It was launched few years back, and much before that another telescope was launched which is called as Hubble Telescope. And you might have seen on the internet that the images of Hubble telescope are good, but they are a bit blurry, especially if we have a cloud in between. For example, there is a galaxy, we have a cloud of dust or even some gases present, so the images are blurry. But when you see the image of JWST, the James Webb Telescope, by the same of the same region, you will get very clear images of those galaxies with no blurring, with no effect of the dust particles. Why? Because the basic difference is that Hubble telescope captures visible radiations, while JWST captures IR radiations. And IR radiations are even better to carry out to take informations with them. They can cross through the clouds of dust and gases and gives you a very clear image. All radiations with higher wavelengths are very useful for carrying information, for transferring information. And therefore we use, for radio communications also, we use radiations which have very high wavelength. So even telescope, which is based on a radiation that comes in IR region, because of higher wavelength, they provides even better images. Anyway, coming back to Raman shift. So Raman shift, in the Raman shift is basically defined as the difference in the frequency of the two incident as well as the scattered lines, and then we plot this Raman shift on one axis, and then we have intensity on the other. Of course, the shift is, although it depends upon the nature of compound, of course, it is independent on the source or the excitation source frequency, because we are calculating the difference. We are not taking the absolute value. The absolute value depends, but the difference always independent of the initial excitation source frequency. As we have in case of IR, we can identify the symmetric as well as asymmetric stretching. Symmetric stretching when the atoms, they are moving in the same direction, and for asymmetric opposite, and bending we have different types of combination which is called as rocking, scissoring, wagging and twisting. Raman, as I said earlier, it is even useful for compounds which you do not have dipole moment, especially the diatomic molecules, for example N2 and O2, since they have two oxygen atom or two nitrogen atoms having same electronegativity, so the dipole moment is 0. In such a case, IR is not useful, these compounds are inactive, so we always prefer Raman spectroscopy. It can also be used for symmetric CO2, because symmetrical CO2 is inactive over there and it can be studied via Raman spectroscopy. Similarly, benzene and CCl4 also we have the same condition, and therefore Raman active bands are there, and we can identify, we can characterize, we can study CCl4 as well as benzene by using Raman spectroscopy. These are the, this is the image of a Raman spectrometer. Again, we are using laser source, laser light, monochromatic light with wavelengths 532 nanometers, 633 and 785. Visible range. The detector against charge coupled devices or photodiode arrays. Photodiode arrays also, they collect the photons, they convert it into electrical signal. Then we have filters which behaves like a monochromator. This is just a working principle where we, from the source the light is coming, we filter it, pass through the entrance slit. This lens is just to turn it. Grating is basically used to break the light into individual pieces, into individual colors. It breaks the light into individual colors, and then finally it passes on the detector. Here it is passed through the sample, and finally it enters, and grating helps to break it into, and it finally falls on the CCD detector or PDA detector, the photodiode array detector. What is the importance or the advantages of Raman? It is a non-destructive technique. Sample remains good. Can be used for aqueous environment, because most of the techniques you cannot use aqueous media directly. If you have a sample in aqueous media, you have to extract it into an organic layer, then only you can use. So the beauty is that it reduces the tedious protocol of extraction. You can directly use the samples in aqueous media. So the sample preparation is quite easy, very useful for organic molecules, inorganics, even biological materials, and quite specific also. Specificity helps you to identify some selective compounds. The resolution of the machine is also good. You can have two peaks quite resolved in a better way. Here it is mentioned the key points, especially for graphenes. In case of graphene oxide, once again I am telling you, from graphite, when we exfoliate, what we get is graphene oxide, which is having lower conductivity because of the presence of COH, OH groups. In order to get graphene, we have to reduce it. The process of reduction does not remove all the oxygens. Therefore, after the reduction, we do not call it as graphene, we call it as reduced graphene oxide. So for that graphene oxide, before the reduction, the D and G bands are at 1350 and 580. These are the position on x-axis, means you get D bands first, followed by G bands, and depending upon the height, the intensity of D and G bands, just by taking the ratio intensity of D bands divided by intensity of G bands, higher this value means higher the disorderness. If the height of D peaks is very high, this shows higher level of disorderness. The same is intensity, ID is intensity of D bands and IG is intensity of G bands. These are the structures. This is graphene, no oxygen. When we exfoliate the graphite, we do not get graphene, but we get graphene oxide. If you do the Raman spectroscopy, you can clearly observe and prove if you have graphene, you will get a sharp peak, only G band, no D bands. But since the method which we are using, we are producing graphene oxide, so we get two peaks, that also not very sharp, the D and G bands. When we reduce it, the reduced graphene oxide has some sharpness, the peaks are even better from graphene oxide. So from the intensity and the ratio, we can identify the disorderness. Conclusion, it's a powerful technique, complement the IR spectroscopy used for compounds which are inactive in IR, especially useful for diatomic molecules or symmetrical triatomic molecules. The role is growing in interdisciplinary areas, in material sciences, biotechnology, DNA. Since it is a non-destructive technique or non-invasive technique, we can use for life systems, in vivo studies as well. Now XRD, the X-ray diffractometry. I told you that here in this technique, X-rays falls on a sample, and just like when they fall, they get diffracted. Depending upon the surface and the planes of the molecule or the solid and the structural arrangement, these X-rays are diffracted at different angles. I gave you an example of a rubber ball. If you throw it on the floor and the floor is rough, it may go anywhere. But if the floor is smooth, it may turn at a sharp angle. And if you throw it 10 times, if the floor is smooth, this angle will be same. But if it is rough, sometimes it may go to that side, sometimes it may go to the other side, or even straight. The same concept, if X-rays when fall on the sample, on the solid, if the solid has perfect arrangement of atoms, they are being diffracted at sharp angles. If the arrangement is not perfect, if the arrangement is not long range, if it is not a good crystalline material, it has amorphous arrangement with no regular order, it may diffract it to anywhere, to broad angles. So then the machine measures those angles. They basically, the diffracted X-rays are being captured. The intensity is red. The angle of diffraction is red by applying the equation, and lambda is equal to 2d sine theta, where lambda is the wavelength of X-ray radiations. D is the interplanar distance between the different planes. Theta is the angle of diffraction. So the machine measures and gives you the theta value. From that theta and this equation, you will be able to determine the interplanar distance. Also from this theta value and the sharpness of the peak and the peak position, you will identify whether the sample is crystalline or not. And on the basis of those peaks, you will be able to determine the particle size, or I should better say the crystallite size of the material, by using a very famous Debye-Scherrer formula. By applying the Debye-Scherrer formula, you can determine the particle size. So X-ray diffraction, it's a versatile technique, again non-destructive, to study the geometry, the phases, the crystallinity of the sample, how the atoms are arranged, the interplanar distance, as well as the particle size. XRD depends on, or it relies on, the scattering of X-rays. When they fall on the solid, they are being diffracted in different directions. And when you have visited yesterday, you have been given the demonstration of two types of X-rays. One was PXRD, powder X-ray, which is very useful for nanotechnologists, the material science. The other is called a single crystal X-ray. The difference is that when we are doing PXRD, we use sample in powder form, and we put a powdered sample. All these dimensions, all the phases are exposed towards the X-rays, because some particles may have X phase at the front, while some other particles may have Y phase, Z phase, because when you are putting powders, there are number of particles. So because of the presence of number of particles, all the phases, angles, all attributes are being exposed. But when we are dealing with a single crystal, there is a provision in the single crystal machine that we have to rotate the sample in all directions, so that all the phases, all the edges, all the angles should be one by one exposed to the X-rays. If we are not rotating, we will get the data from one side of the sample only. So for single crystal, we have to create a crystal first, and then we rotate it to expose all the these phases and angles and edges to the X-rays. And then we use the Bragg's equation, which I have just mentioned, and lambda is equal to 2d sine theta. So when a monochromatic light of X-ray interacts with a crystalline material, note it down, the crystalline material, the atoms, they act as a scattering center, and they scatter the light with some sharp angles. And from there, those angles, we can identify and draw a diffraction pattern, draw the structure, the morphology of the sample. This is a solid. You have been shown here the different planes. When the X-ray falls on the front, the first layer plane, they are diffracted in one direction. Since the distance is constant in all the planes, the sample is crystalline. Again, you can see the angles are same, and they are being diffracted at sharp angles, same angles. By measuring the sample, by measuring the angle, and based on the lambda and n values, we can determine the D. Again, the instrumentation in all spectroscopy, the two most important are X-rays and detectors. We also have sample storage and goniometer here. This is the X-ray tube and a goniometer which helps us to rotate the sample, and from this tube X-rays are being generated. Here we put the sample. On one side we have a connection for incident radiation, on the other side we have detector. When X-rays are falls on this object, on the sample over here, they are being diffracted. Some are going straight transmitted, while some of them are being diffracted. These diffracted X-rays are captured by the detector, and the angle as well as the intensity is calculated. And you can see that the angle, the sample is a little bit tilted because of this goniometer. We are rotating, we are exposing all the dimensions, all the faces. So X-ray tube, the sample, this is scattering, and again we have a detector. This is the schematic diagram. This is the actually, this is also even a schematic diagram. This is a flow chart. You can see how the different parts are connected. For the machine which we are using and which you have been given demonstration, the lambda is 1.54. We use copper K alpha radiations with a wavelength of 1.54 angstrom. The goniometer, as I said, it is used to precisely control the angle of the sample and the detector. We can rotate it, we can change the angle, we can change the platform, we can tilt it, keep it straight, just to expose all the dimensions. We can make measurement of theta or 2 theta. So the main purpose of goniometer is for the sample alignment and rotation. Then we have detectors. There are different types of detectors possible. Scintillation detectors. We generally use solid state detectors here to check the intensity as well as the angle. These are some samples which are being prepared for the analysis. There is a sample holder, we put the sample over there and do the analysis. And this type of curve is obtained, this type of plot is obtained. You can see very sharp peaks. This shows the crystalline behavior, and from these peaks you can also identify the crystallite size using Debye-Scherrer formula. If these peaks are broad, this shows the material has low crystallinity. I think I am running out of time. These are the application used for studying the morphology, the crystallinity, the particle size, even orientation studies also. If you have any question relating to XRD or Raman, I will try to answer, and then Dr. Asfar will be explaining you little bit about Raman spectroscopy, the application part, just for 5 to 10 minutes. Then we will proceed for lunch. Thank you very much. कुछ लगाना है? ठीक है, घंटा खींच के आराम से. एक बजे से 2:30 तक है कि हम लोग चलेंगे, तब नमाज को चलना, और गर्ल्स वगैरह का लंच स्टार्ट करा देंगे. ठीक.
Working.
भेज दिया है मैंने आपको. किस पे किया है? देखिए बारिश यही तो है, रास्ते में रहेगा क्या?
Okay, we have very short time. In very quick time, I want to discuss some experimental results of graphene. Actually, you very familiar to the word graphene, reduced graphene. So I want to share some experimental results, and this is our paper, research paper of my PhD work. So basically, the application which we done in this paper is the removal of uranium ion. Uranium ion. This is the actually our supervisor has some research project related to the radioactive material removal of bark. So we use graphene oxide, reduced graphene oxide as an adsorbent. So what we want to share here is some results. So first of all, I want to show some tentative scheme. What we discuss in this paper is that this is a graphene oxide, as you see here, and we use some linkers and nucleotide AMP, and this is a GCC dicyclohexyl amidamide, and this basically the linker which convert the carboxylic acid into the amide group. So we fabricate our graphene oxide with this AMP using DCC as ethyl glycol as a linker. So we got RGO AMP. This is RGO means reduced graphene oxide, AMP because we use AMP. So we give the name AMP. So now come to the results, which is very important to understand. So this is not a slide, this is actually paper we directly discuss here. So first of all, come to the FTIR plot, FTIR data. So the red one is the graphene oxide. So this in graphene oxide you say is a 730. So this 730 is the signature peak of which functional group? It is a signature peak of carboxylic acid, near about COH. This is a peak of C double bond O, the COH of the carbonyl group of the COH. And this peak around 3440, this represents the signature peak of hydroxyl group of COH. When we convert it into RGOMP, when we reduce this, so what happened? We observed different types of extra peaks around 3328. Can anyone tell this 3328 peak represents the NH peak, the stretching peak of NH, because we convert the carboxylic acid into amide. So we have NH peak. This 2933 and 2840 is basically the peaks of the CH peaks, strong CH peaks. And this 627 is basically the carbonyl group peak of the amide, 627. And you feel that this 1730 peak is disappear in this RGOMP. It means that our carboxylic group is mostly converted into the amide group. And we also confirm this experiment by using a specific titration called Bohm titration. Bohm titration. Many of you, I think, listen this first time. Bohm titration. This is a titration in which we can calculate or we can quantify the concentration of the acidic functional group which is present on the graphene. So it is not a time to define this. So this 1627 is the peak of the amide group, and this 1575 is the peak also related to the peak of the amide. And this 1230 peak is of the phosphate group, because AMP is a nucleotide, it has a amide group also and also phosphate group. So this represents, so from this we can conclude that we have a very good fabrication. We can reduce the R graphene oxide into RGOMP. Now come to this one. So this is a Raman spectra of the GO and RGO. Now you can, people, I think you can easily interpret this. You can see that in case of GO, the ID by IG ratio is 0.859, and in case of RGOMP, the ID by IG ratio must be increased or must be reduced? Must be increased. And what we get in the result? 0.880, because when we reduce our graphene oxide by AMP, so what is the meaning of this? We increase the D band. We develop the defects in the RGOMP in the basal planes, so that the intensity of D get increased. We increase the wettability, as Professor Leo discussed in the very previous lecture of this GIAN program, the wettability. So it enhances the wettability. So we get ID by IG ratio in case of RGMP get enhanced. So when we reduce it into the, so we can make our RGMP a very good adsorbent. Now some results I want to show you, not all, but a comparative table I want to show. This is the XPS result, very normal, nothing like very new in this. But I want to show a comparative table of this. What is the beauty of this RGOMP? Look this. Look this. This is a comparative table what we do. In RGOMP in last 23 number, 23 CL number, the RGOMP, we get the Qmax value, Qmax, which is the adsorption capacity, mg per gram, is 3024. Means 1 gram of RGOMP adsorb 3024 mg of uranile. And uranile UO2 2 plus is itself a big molecule. But it enhances the surface capacity. So we get this result of adsorption capacity of 3024 in a very quick time of only 4 minutes. So you can see, you can check or you can absorb the surface activity of the graphene. In just 4 minutes you can get the very high adsorption capacity of 3024. So from this what we can say that if we functionalize our graphene into another functional group by using another functional group, we can use our graphene oxide in different applications. As for example, this is the one example in which we use uranium, People can use this in other heavy metals like arsenic, lead. So those you people, if you want to work on this application part of the graphene in the research, so you can work on this because there is a very scope. Nowadays, for example, in India, when I was in some lab, there is a very big problem of arsenic in UP East, mainly in UP East. So we have to think like this, we have to approach like this our research so that we can prepare some type of absorbent like this which is cost effective also, which is very good reusability, we can use good recyclability. And one more result I want to share also this, our material has very good recyclability also. We can use this up to 5 cycles and we can get the adsorption capacity. This is the theoretical, it is experimental absorption capacity around, I think this one is around 150. So we can lose only around 30 to 40 absorption capacity, 30 to 40% of this, and we can get up to 5 cycles. So so what I want to say that if you want to work in this metal chemistry, so you have to prepare that type of absorbents, that type of the material which has good recyclability, or most thing is that you have to prepare more economical also. How you make economical? You increase its recyclability. So that you can use up to 10 cycles, up to 15 cycles and commercialize it. And main thing is the commercialization. So this up to the results which we want to discuss here, because application part is very important. How we use the graphene in the water treatment. So anyone have a question related to this? Because it take already, we are 5 to 7 minutes over. Very quick. Quarter past. Anyone? This is actually this is the basically the tentative scheme which we prepared by after characterization. What we think, because we not perform any DFT studies in this so that we can confirm this, but what we think and what we get from our FTIR data and from Raman and from XPS, that basically what AMP and DCC, AMP is basically the nucleotide and DCC is basically the linker which convert the carboxylic acid into the amide. It is a very good linker. You just put the DCC in the sample in a millimoles, you calculate the millimoles and put into the into the round bottom flask and go for 12-hour reaction. You get a very good results. So this is basically, and this structure you can easily differentiate from the characterization part. For example, you can easily confirm from the FTIR, and you can also, we also perform the zeta potential. The zeta potential of, you can see the red one is graphene oxide. You see that when we perform our zeta potential from 2 to 9, So you can observe that although pH2 is very acidic, it means very high concentration H plus, but in that case, in that pH also, our graphene show negative zeta potential. But when we same apply for the reduced graphene oxide, we can get the zeta potential in positive part. It means that we have some functional group which can acidify, which can easily acidify at this quotient. Therefore, we get the 0-point charge at 2.31. When we cross this pH, we get the negative values. This is also the difference which we observed after the modification. Actually, we basically we confirm, we take this, we observe this, that the COH group which is present at the outside the basal planes of the graphene oxide. Actually, we show, this is, write this, read this, representing the, it is a tentative sites. It is a tentative sites. You confirm it from some theoretical studies like DFT, I think. Anyone? Functional group के लिए डाटा it is fixed. It is fixed. लिंकर मतलब जो लिंक कर दें, which link two functional groups, है ना? Anyone? I already told you that we go up to 5 cycles. We go up to 5 cycles. So you can optimize more. It is up to your hand how you produce your material. Very good question, because uranium has a tendency, for example, some elements have a tendency to bind particular another elements. For example, if you found iron, if there is, for example, in geological terms, if we have iron, so it is very probable that you found arsenic here, because arsenic has a very strong bonding with iron. Similar in these cats, uranium has a very strong bonding with phosphates and nitrogen. Therefore, sometimes you found some studies that where there is phosphate rocks are found, you get uranium also. When you check some studies in Haridwar side, because there is a Ganga, so in that there is a very rich phosphate rocks. So in some studies, it confirmed that we found uranium in the, because uranium leach out the system and comes into the groundwater. So therefore, we use this concept and modify our material by using phosphate groups on the one side and nitrogen on the first, because nitrogen has also very good compatibility with the uranium. So we have two binding sites, one for the nitrogen, one is the phosphate. In that, uranium becomes in the middle, so uranium binds with those, with phosphate also and with the nitrogen. Therefore, we get very high absorption capacity. With one molecule, you have to calculate with this, with the millimole formula. This is nucleotide. You can easily purchase from the market. It is an available compound. बिल्कुल कर सकते हैं, बिल्कुल कर सकते हैं. आप कोई भी सिंपल फॉस्फेट ग्रुप ले लीजिए. आप for nitrogen source, you can use amino acids also. Now it's time for lunch, and we can meet on 2 o'clock. At 2 o'clock sharp, please come and assemble here in the conference room. And there is an exam.
Please come downstairs. Waziha, Ghania, please come here. You have to submit the feedback form along with this questionnaire. So please complete the feedback form as well as this MCQ question paper. Question paper. आपको नहीं मिला है? नहीं, नीचे बचे हैं बच्चे. आप बीच में चले जाइए. The time starts now. 30 minutes का पेपर है. लिखा है. 1 घंटे का पेपर है. ऑफलाइन ना 60 minutes. ऑनलाइन कम क्यों दिया? चीटिंग कर रही है. Everyone is requested to switch off the mobile phones. All the questions are compulsory. आप लोग नीचे आ जाइए. ऊपर जगह नहीं है, आप लोग नीचे आ जाइए.
For your kind attention, only 15 minutes are left and you have to submit your question, the answer sheet at 3 o'clock. You are also requested to submit the feedback form. These are the requirements that has to be met, then only your certificate will be processed. At 3 o'clock, we'll be starting the final lecture cum tutorial in which Professor Hetao will be explaining you the methods of fabrication of DNA nanoparticles, and he'll be showing you some videos of his own lab where his students will be designing different DNA nanoparticles. So, first of all, submit your copies on time, the feedback form on time. Thank you. Keep quiet. Please submit your copies. You have only 10 minutes. And those who have not yet collected the feedback form, please collect from the organizing team, fill it up, and return back. Feedback form is very important. All of you have submitted your copies, or anybody is still doing? Aisha? All have submitted over there? Done? Everybody? ठीक है. Count all the answer sheets. We'll be starting in 5 minutes, the next lecture.
First one, that's after the D.
First, second.
Okay, the first way. I share entire screen. Then we will open. Select entire screen. We will choose.
I think we start at the 3. So.
Okay.
कितने हो गए असाइनमेंट? वो कितनी कॉपीज है? 87. सुहेल ऑनलाइन चेक करो कितने आ गए. इसको लिखो, नंबर लिखो ताकि भूलो ना.
एक मिनट, मतलब मैं शेयर कर दूं उसके बाद.
कॉपी. Please pay your attention. Welcome back. You have now just completed your test. You have just appeared in exam, an MCQ based test. The mandatory requirement for GIAN. Now we are starting, I'm very sad also, the last session of this GIAN program of the 5th day, the last tutorial which Professor Hitao, he has initiated. Now he is doing the, from Agaz to Anjam, he'll be taking the last lecture. In this lecture, he'll be explaining you about the preparation and characterization of nanostructures, the DNA-based nanostructure, and how they are actually being done in his own lab. So I welcome Professor Hitao Liu. Please give a huge round of applause.
Thank you. All right, so in the next 20 minutes or so, I want to share some practical aspects of DNA nanotechnology, and in particular, I asked one of my graduate students recorded a video of the whole process that we used in our lab, making the DNA triangle nanostructures that we have used in many of our experiments. You may recall in the second lecture today, I showed a lot of experiments, and a lot of those are based on this triangle structure that we have been using since 2010. Just to give you a quick refresh of what this is one of those structures. Let me turn on the pointer. So this structure is created. So just to give you a refresh, this is the structure created first by Paul Rothman, the guy at Caltech, who in 2006 invented the origami technology. This is done by mixing one scaffold strand, the MP13 bacteriophage DNA. The length of this DNA is about 7,000 bases long. Again, we know this sequence, so we use the computer program to come up with about 300 of DNA staple strands. Okay. And when we order this, this is coming from a commercial DNA synthesizing company called IDT, Integrated DNA Technology. And we essentially got 300 small test tubes, 300 of those tubes. Now, the unique thing about this origami approach of making DNA structure is it's highly robust in the sense that we do not care too much about the stoichiometry. So if you think about your typical, most of the organic synthesis or inorganic synthesis process, you really want to make sure, oh, I add exactly the right amount. Okay. That's not the case here. So instead, what we do is we have all the 300 different kinds of short staple strands. We mix them together at roughly 1:1 ratio. Okay. Sometimes the concentration is not quantified very well when you purchase this DNA from commercial companies. They just do a quick UV-vis quantification, and sometimes they don't even do purification. Actually, we can use DNA strands that are not purified at all, just fresh out of the solid state DNA synthesis, no purification, no HPLC, no nothing, and very crude mixture. And we can use that, and the synthesis would work out just fine. So all the staple strands are, 300 of them. We mix them into one stock solution. And all the staple strands individually, they are roughly 1:1 ratio. Okay. And then we have the scaffold strand. This is a very long, 7,000 base pair long strand. And we usually make sure that the staple strand overall are in excess. So you can be as aggressive as 1 scaffold to 10 staple strand, but it can be as low as 1:2 ratio. As long as the staple strand is more or less in excess, you're fine. Sometimes we even did 1:1 ratio, and that turns out to be fine as well. So again, this is a highly robust process. It does not care about stoichiometry that much. Okay. And the reason being, you have so many ways to fix the problem. Because you have 300 staple strands, imagine if one of them is not enough, it's okay because you already folded the structures 299 times. The missing one it's not going to create too much a problem. Okay. So that's the reason this self-assembly process is not sensitive to the specific and exact stoichiometry, and that's a very unique feature of this, a very beneficial feature of this origami approach. Okay, so that's our stock solution. And then we make additional buffers, and you may notice the student specified pH 8, and this turns out to be important. Now, first of all, the stability of DNA nanostructure certainly depends on pH. Now, what's more important in this case is when we deposit DNA nanostructure onto silicon, which is our substrates, the deposition, the interaction between silicon and DNA nanostructure heavily depends on pH as well. DNA carries a lot of negative charge because of the phosphate group. You may recall a question in the morning, what is the interaction between DNA and the silicon oxide surface? That is by ion bridge mediated by magnesium 2+. And to have that ion bridge interaction, you have to ionize silicon oxygen, the silanol group, the silicon wafer. And that requires a high pH. So in this case, we used a pH 8, and that just worked out well. And if we reduce the pH, oftentimes we see reduced adhesion of DNA structure onto silicon wafer. You also notice specific reference to magnesium 2+ in this figure. Again, that is very important because when you make double helical structure, you are packing a lot of negative charge into a small area, small volume. DNA has this negative charge backbone, and you want to neutralize that kind of negative charge. And the best way to do that is to add a divalent ion, magnesium being one of the most often used. Okay. As I said just before, magnesium 2+ also plays this role of ion bridge to mediate the adhesion of DNA structure to silicon wafer. Okay, so once we have everything, we mix all this stock solutions in with the appropriate concentration ratio. And usually, just for convenience, we distribute the solution. Usually, this will be in a 1 ml or 5 ml Eppendorf tube. We put them into smaller ones because later step we need to do centrifugation. We need to do a lot of things that requires mass balancing. So usually we split up into 4, 2, 4, 6 smaller tubes. And then we put it into a PCR machine. And what we're doing here is we're going to heat up the temperature above the melting temperature of all the double helical strands, sections. Again, here we have about 300 different strands, and each strand is about 20 to 40 bases long. Okay. We heat it up, first of all, to about 95 degrees Celsius to ensure complete denaturing of all the strands. And then we slowly cool it down. Now, the cooling rate is very important here because you want to give the system enough time to explore all those different reaction pathway. If somehow it got trapped in the wrong intermediate step, you want the system to have time to come back. So it's important to do the cooling slowly. Depending on the particular nanostructure you're making, this can go as short as a couple of hours to as long as 2 days. Again, the slow cooling would allow the system to get out of the trap state and come to the final thermodynamic minimum, which often is the nanostructure we're looking for. Okay. So once we're done that, what we end up with then ideally would be a lot of DNA nanostructures. And if you keep in mind, our staple strands are in excess. Okay, so we already have anywhere from 2 times access to 10 times access, depending on our particular mode of the day, and that actually fluctuates quite a lot. So at this step, once you cool everything to room temperature, you're going to have a lot of DNA structure, but also a lot of staple strands floating around. So ideally you would consume all the scaffold strands, but because you have an excess staple, so you need to get rid of those excess staple strands by some kind of purification. And in this case, we have some structures that are pretty big, 100 nanometer in size. This is the edge length of the DNA triangle. Okay? And we also have a lot of single-strand DNA, essentially each about 20 to 40 bases long. Those are much smaller. Even if you are not considering the single-strand DNA are in random coil structure. Okay? Each base is about 0.3 nanometers if it's a double helical DNA. And if it's random coils, the effective size is going to be even smaller. So if it's double helical structure, 40 bases long is about 12 nanometers in length. But if it's a single strand, it's going to be random coil. It's going to be a random globular thing. That's probably a couple of nanometers in size. Okay. So what we do here, then we need to separate things simply by size. There are a lot of ways you can do the separation in molecular biology. You can easily separate biomolecules by size using gel electrophoresis, and that can certainly be done. But it just takes a longer time, and more instrumentation involved. You have to make a gel, and you have to stain the gel, and you have to extract the DNA extract from the gel. It's a lot of hassle. So the easier way to do this is to use a filter. And what you see here is this special filter. And at the bottom of, you see this is a 2-compartment filter. Okay. On the top part, we will add the DNA nanostructure raw reaction mixture to the top. And at the bottom here, there's a membrane that is size controlled membrane. And you can pick a particular molecular weight cutoff. But because the size of the two components are so different, it's pretty safe as long as you pick something meaningful. Okay. So I think we really pick a molecular weight cutoff around 100,000 or something like that. And then in this configuration, the small species will go through. The DNA structure will stay on top. Okay. So to force the filtration happen, we centrifuge the whole thing at about 6,000 RPM for about 5 minutes. And we have to do this multiple times because we do not want to centrifuge everything to dryness. That will result in the DNA structure being stuck to the filter. So oftentimes we would centrifuge to about 1/3 of volume left. Then we dilute whatever is left with buffer and do it again. And then centrifuge again, dilute it, and dilute again. We usually do it 3 to 5 times depending on how much DNA, single-strand DNA do we want to get rid of. Okay. Most of the time, 3 times is sufficient. But in some special cases, when we really want to lower the concentration of single-strand impurity, we do it 5 times or more. Okay, so after that, we will collect whatever is left on top. Again, the filtrate contains the single-strand, excess single-strand. But the remaining liquid here contains DNA structure. As you see, there are triangles here and there. So we take those, and then the next step is we're going to do quantification. This is a standard technique everybody uses in chemical biology. DNA has a UV absorption, and then we simply do a quick UV-vis absorption measurement. And from the absorption data, we can quantify the concentration of DNA nanostructure in the solution. Once we have those, we usually break them apart into even smaller tubes so that we can store them in the fridge. If we store in 4 degrees Celsius, this usually survive at least a couple weeks, maybe even longer. We didn't push that limit very hard. If it's a room temperature, at least a couple of days, maybe a week. Again, we usually use them right away, but storage in fridge 4 degrees Celsius is not a problem. There are also a lot of research done to analyze and understand how this DNA structure would react to frozen conditions. And the general message there is by freezing the buffer, you will do some damage to DNA structure, but not all. So there are also tricks to protect the DNA structure from the mechanical damage that occurred during the freezing process. And after that, we deposit the DNA structure onto a substrate. It's usually silicon because it's flat. A lot of times we also use mica. Again, this is a very flat substrate. The height of the DNA structure is about 2 nanometers, so you really need something that is super flat at the atomic scale. And silicon wafer and mica are the two most often used substrates. In both cases, there's ionic interaction between the DNA structure with the substrate, and we'll be able to use atomic force microscope to image those. And so this is the whole process, and I'm going to show a video that essentially goes from step 1 all the way to step 10. I'm going to pause the video to explain what is what, so hopefully you'll get a more intuitive idea of, in the research lab, how this was done. Okay. And by the way, this was taken from a qualifying exam presentation of my student. His name is Shubhanka Kundu. And he also took the video that I'm going to show you in a sec. All right. Okay, the person in the video, her name is Katie. She is an undergraduate student conducting research in my lab. She has been working with Shubhanka in the past 2 years or so, working in the area of DNA nanotechnology, broadly defined. She is standing in my lab. As you see, my lab is very disorganized, a lot of chemicals sitting around. But on the positive side, she has gloves. That's good. Chemical hygiene protection. She already has her glassware, so, and because she's working with ecosystem, I thought, okay, that's fine. And I'll play this and I'll pause from time to time. Most of the steps is pretty self-explanatory. Okay, but if you have any questions, feel free to interrupt me. Audio is gone.
And then we have pre-prepared DNA staple strands. And we combine those two in a 10-to-1 ratio, so 10 times the amount of staple strands. The other two things are water and some buffer, scaffold DNA or...
Okay, so I'll replay from the beginning. And as you see, she's explaining everything. So hopefully I don't have to interfere too much.
Origami nanostructures. So it's a mixture of four things. First, we have DNA scaffold strands that we buy.
Now that scaffold strand, that is already a mixture of 300 different DNA scaffolds into one. So when we bought those scaffold strands, we have a pack, this array of Eppendorf tubes. And when I started, I don't have a lot of funding, so my student pipetted one after another 300 times to make a stock solution. But fortunately, we don't have to do it again because we already did it 15 years ago. We're still using that mixture until today.
And then we have pre-prepared DNA staple strands.
So this is a long, very long strand.
And we combine those two in a 10-to-1 ratio, so 10 times the amount of staple strands. The other two things are water and some buffer. And we usually combine those in a tube just like this using some micropipettes. And once the solution is mixed together, we divide them into 4 equal allocates into tinier tubes like this.
Okay, another reason we use the smaller tubes is because those are the standard. I think I'm not a chemical biologist by training, but I think those are the standard tube size for PCR machines. And because we use PCR machine for the annealing process, which is just convenient, we move them into the smaller test tubes, Eppendorf tubes.
And then we take them to our PCR machine. Alright, so now that we have our 4 tubes of DNA solution, we bring them to our PCR machine, and we put them in the little slots right there. And then we get to select what type of nanostructure that we want to make. So there's a bunch of different kinds, but you just select it, press start. They can take anywhere from 2 hours, even up to 48 hours. And once that's done, we take all 4 tubes and put them into one centrifuge tube. Now these tubes actually have a filter in them.
Okay, so that's the size selective filter I was talking about. So as you see in the video, this part was placed on top of another Eppendorf tube. So the filtrate will naturally go down, and whatever is left would be would contain the DNA structure. And also another feature I want to mention is in some experiments, you will eventually be limited by the power of the PCR machine, because in some cases you have to do it really, really, really slowly. And in those cases, sometimes you have to be inventive and creative and come up with some kind of apparatus yourself. For example, a big styrofoam box so that you can cool it really, really slow. Because not all PCR machines will allow you to do 2 days or even a week of slow cooling. Sometimes it just doesn't exist. Okay. But in most of the cases, as she mentioned, it could be as short as a couple of hours. But we have done some other experiments where 2 days of slow cooling is required.
If you can see that. And as the solution gets filtered through via centrifuge, so just go like this, only the DNA staple strands come through, while the DNA triangles, or whatever nanostructure we make, stays in the top section. And we add buffer as needed just to make sure that the solution doesn't run dry. And once we filtered it through, then we take it to our UV Vis nanospectrometer machine to check the concentration of DNA in solution.
Okay. All right, so another thing I want to mention about the purification process, because we do not want to filter, have the DNA nanostructure solution being filtered completely dry. So if you reduce the volume, if you centrifuge down to the volume of original 1/3, and then you refill buffer to the original volume, you're gonna reduce the single strand impurities strand, their concentration by a factor of 3, right? Because original volume is 1, you essentially remove 2/3 of them, and then you refill. So if you do this once, then you're going to reduce the impurity concentration by a factor of 3. If you do it twice, that would be a factor of 9. If you do it 3 times, that would be a factor of 27. Now, you may think this is quite a lot of purification, but you should also keep in mind that we're using huge excess of staple strands. As Katie mentioned, in this particular case, she's using 1 to 10 excess of DNA single strand, the staple strand. So ideally, after the reaction, you have 1 to 9 concentration, being 1 being the DNA nanostructure, 9 being the excess single strand DNA. Okay. So if you do the purification twice, you will get concentration ratio of 1 to 1. So from that perspective, your reaction mixture is still very, very dirty. So in this context, you need to do the purification 4 times so that you can reduce the concentration of impure single strand to about 10% of that of DNA nanostructure. If you do it 5 times, then you can reduce it to about 3%, and that would be something I consider adequate, 3% impurity. Now, you can also, think about this at the very beginning of making this nanostructure. Instead of doing 1 to 10 ratio, maybe you can use 1 to 3 ratio, and that will change the calculation completely, and then you don't have to do the purification as many times. All right, so I think the next step she's going to do
our UV Vis nanospectrometer machine to check the concentration of DNA in solution. Okay, so to check the concentration of DNA in the solution, we go to our spectrometer machine and open the software and hit nucleic acid. And once that loads, it has to do its own verification routine. And then at the top, we follow the instructions. So our blank solution will just be water. So I'll take 4 microliters of water.
So the instrument he used is a microvolume UV-Vis absorption spectrometer, and what you see here, so this part, let me see if I can draw it. So this part, there's a sensor there. You can add a drop of water droplet onto it, and you push this part down, and that water droplet will form a meniscus, and it only requires a couple of microliter volume. So it's very economical in the sense that you are not wasting a lot of your sample. We also have other instruments that require the more conventional cuvette, but that requires much more volume of your sample. Now, when you think about the kind of volume we're dealing with, usually it's not even more than a milliliter. So, so we prefer this kind of micro scale measurement. Let me clear everything.
And I'll put it right on the tip where the sensor is. So now we have just a tiny little.
All right, you may see this is a small water droplet on top of the light sensor, and when the top of when there's this cap come down, it will form a defined gap, and from there you can quantify the concentration.
Droplet on there. We close the machine. And now that we've done that, we hit blank to measure the blank. All right, now it's saying we can load our sample. So first we're just going to use a Kim wipe to wipe up all the water so that it doesn't interfere with our concentration measurement. And so now I'm going to take a sample that we've already prepared and filtered. I'll take another 4 microliters of that and put it in the same place as the water. All right. We close the machine again. And now we click the big green button. And so our goal after we create a sample is to have a concentration between 20 to 50 micrograms per milliliter. And so as you can see, right here in this green box, the concentration is 46.8.
Okay, so you may find this is a familiar shape. If you have taken chemical biology or biochemistry, this is a familiar shape of UV absorption of DNA, and you can quantify the concentration by looking at absorption at 260 or 280 or a combination of them. My memory is rusty, but the number here you can see A260 and A280. Those are two important numbers that chemical biologists usually use for quantification of DNA. From the absorption, we can calculate the concentration, and we want a certain range of concentration because most of our work are done by via surface supported, substrate supported sample. So we want to get a reasonable surface coverage. We don't want it too high or too low.
Which is just fine for us to store in a fridge to use later. Now, if the concentration was too high or too low, we would just take the solution back to the centrifuge and either centrifuge it more or just add some more buffer until we get the concentration that we want. Back in the lab, after we check the concentration of...
Okay, so now she's sitting in our shared facility and she's operating an atomic force microscope. And she's gonna show the sample loading and the data collection process. I believe the sample she's investigating is on a silicon wafer substrate, or could be mica, but one of the two. So I'll stop and explain what the machine is in a sec.
Our nanostructures, we would take them and put them onto some type of substrate, let that deposit, clean it off, and then bring it down to our AFM room. So here's some examples. This one right here is a silicon wafer, and the rest of these are mica. Those have our DNA nanostructures on them already. And then next, we have to set up an AFM tip. So here's a box of these tips, fun fact. This whole box is $2,000. So we have to be very careful with how we handle them. That's why right here we've already got the tip set up in...
So this certainly depends on the manufacturer of AFM. So different model of AFM have different procedures to load sample, to load tip. So this particular model is a Xylem MFP3D. And it's a pretty good instrument for standard topography imaging, but still is a 10 years old model. We have been using, you see the Asylum Research, that's the manufacturer of this AFM. And this is their tip loading setup, I believe.
In here. And once that's ready, we can take our sample and the tip to the machine. So how does the AFM machine actually take an image of our sample? Well, let's imagine my hand is our sample, and this is a DNA nanostructure. And so the tip that we talked about before would be lowered onto the sample, and once we start taking an image, it vibrates like this and moves along the sample. And once it gets to some sort of structure, it moves around it because it doesn't actually touch the sample, but just hovers right above it. So if there's anything in the way, it has to go above it. And that's how the instrument actually takes an image.
I want to make a small correction. It's tapping, but it's actually in a non-continuous contact with the sample. There is actually a non-contact mode of AFM imaging, but that's not the imaging mode we're using. We're using the more common tapping mode imaging, vibrating the tip at about 300 kilohertz in our particular setup, and having the substrate moved up and down so that the amount of tip sample interaction is always kept at a constant. So this big box is the AFM sample chamber, and inside is the scanner and the sample holder and everything. There's electronics outside the box. As you see, there's a lot of wires and stuff. The whole box can also be purged with nitrogen. As you see, there's some kind of gas cylinder regulator here. In this case, we are not particularly concerned about the atmosphere. but it's possible to purge everything under nitrogen, for example, if that is needed.
And because the tip is so sensitive, it's very sensitive to vibrations and sound, so it's in a fancy little box.
So a little bit more on this instrument. The whole thing is put on a vibration isolation desk. But even on top of that, you see this big chunk of rock, just to make it more isolated from building vibration. And this whole lab is also on the first floor of a very old building. So the vibration is minimized in the first place. You also see the inside, a lot of aluminum foil and things like this. That's designed, I believe, to reduce thermal fluctuation because we are measuring a lot of dimensions at the nanoscale. So that's 10 to the minus 9. Now, if you think about the thermal expansion and contraction of a lot of materials, changing the temperature by 1 degree or even less can produce a lot of thermal contraction and expansion. So you have to create a very stable environment in terms of temperature, airflow, and vibration, certainly, to get high quality imaging.
So we would take our sample and put our sample onto the glass slide here.
Okay. So as I said, the way it works is the tip will scan the surface. Now that's the easy way to explain it, but in reality, what happens in this model is that the tip actually doesn't move. The sample moves. Okay. So the tip stays at the same location. There's piezo, so you see this is a sample holder, and there are piezo drivers in both X and Y dimension, and also actually Z. So when the tip vibrates, it just vibrates its stationary location, and the sample moves back and forth in a 2D scanning pattern.
Then we take our little thing with the tip and put it into this part of the machine.
And this part, that's the scanner. And it holds the tip, and she will very soon put the scanner, so right now it's upside down, and she will invert it so that the tip will be facing the sample, and then she'll do other things to make sure that tip eventually will contact the sample.
To make sure it's secured, then we take this very carefully, put it on top, and so now the wafer and the tip should be right on top of each other. And once this is set up, we go back to the software to set a few parameters. So after setting those parameters in the software here, we close the door to the machine, press go, and the image will show up in these boxes over here. And so this would be a 20 micrometer by 20 micrometer square picture. And once it shows us that, we can zoom in and out in different locations, which will end up looking something like this.
Okay, so she's not showing the real-time operation. So in a real operation, you would need to first load the tip, approach the tip to the surface. The tip is vibrating all the time. And as the tip is getting closer, the degree of vibration would feel being interfered. And the machine can detect that. And once it detects this smaller vibrational magnitude, the machine knows, oh, it's hitting the surface. And once that happens, it will stop pushing even further to the surface, and instead using the piezo in the Z direction to maintain a constant tip sample distance. And then the scan will start. Some important parameters we typically use, one is the scan rate. This is how fast you scan in one back and forth. Okay? And in a typical experiment, we usually do a 1 Hertz scanning. This is actually pretty slow because you go here and then go back. So let me draw this. So what does this 1 Hertz mean? It means I go this direction and come back. That's 1 second. Okay. Now, each one of those images, you'll also see other parameters, scan lines and scan points. And those are 256. So what this means is each image here is made of 256 lines. And in each line, the machine takes 256 data points. So the resolution of this image is 256 by 256. Now you can increase that resolution or decrease it as your experiment would require. Usually we do 256, 512 is also common. You increase the resolution, but you will pay price in time, because if it's 1 Hertz scanning, then each image will take 256 seconds to be collected, and that's about 4 minutes. So a lot of the time of my students spend on sitting in front of the computer and evaluate the sample and find the correct sample area, so on and so forth.
So that was 20 micrometers. This is a 3 micrometer section. And here are our DNA origami triangles, which are just one of the structures that we're able to make. And so you can see this triangular shapes here, 3 straight sides, and you can see the whole.
So the important feature we look at here is the triangle, but also there's a hole in the middle. And this particular structure is very, very useful for a lot of our lithography work. You may wonder why we have been using this triangle structure for so many years. And the reason is because it's very, very good for that kind of work. So in this seemingly very simple structure, there are a number of features that are very useful. One is the sharp edge. Another is the hole in the middle. Okay. And even actually there's additional smaller holes on the edge of the junction of the edges. So all those features would allow us to make a very quick and qualitative quantification and understanding of the quality of pattern transfer. That is the most important parameter we are trying to understand and improve. So for that reason, we've been sticking to this triangle structure for a long time, and we're most likely going to keep using them in the foreseeable future.
In the center of the triangles. And what this tells, this lets us know is that the structures were made correctly. However, you can also see some of them aren't quite perfectly triangular. Maybe some of them don't have a hole in the center, or maybe the sides just came apart. And that's what we're able to check in the AFM image.
Okay, so that hopefully gives you a general sense. Sure. I'll play again without interruption so that you have a more meaningful understanding of what's going on.
Hey guys, today I'm going to show you how we prepare our DNA origami nanostructures. So it's a mixture of four things. First we have DNA scaffold strands that we buy. And then we have pre-prepared DNA staple strands. And we combine those two in a 10-to-1 ratio, so 10 times the amount of staple strands. The other two things are water and some buffer. And we usually combine those in a tube just like this using some micropipettes. And once the solution is mixed together, we divide them into 4 equal allocates into tinier tubes like this. And then we take them to our PCR machine. Alright, so now that we have our 4 tubes of DNA solution, we bring them to our PCR machine. and we put them in the little slots right there. And then we get to select what type of nanostructure that we want to make. So there's a bunch of different kinds, but you just select it, press start. They can take anywhere from 2 hours, even up to 48 hours. And once that's done, we take all 4 tubes and put them into one centrifuge tube. Now these tubes actually have a filter in them, if you can see that. And as the solution gets filtered through via centrifuge, so we'll just go like this, only the DNA staple strands come through, while the DNA triangles, or whatever nanostructure we make, stays in the top section. And we add buffer as needed just to make sure that the solution doesn't run dry. And once we filtered it through, then we take it to our UV Vis nanospectrometer machine to check the concentration of DNA in solution. Okay, so to check the concentration of DNA in the solution, we go to our spectrometer machine and open the software and hit nucleic acid. And once that loads, it has to do its own verification routine. And then at the top, we follow the instructions. So our blank solution will just be water. So I'll take 4 microliters of water, and I'll put it right on the tip where the sensor is. So now we have just a tiny little droplet on there. We close the machine. And now that we've done that, we hit blank to measure the blank. All right, now it's saying we can load our sample. So first we're just going to use a Kim wipe to wipe up all the water so that it doesn't interfere with our concentration measurement. And so now I'm going to take a sample that we've already prepared and filtered. I'll take another 4 microliters of that and put it in the same place as the water. All right. We close the machine again. And now we click the big green button. And so our goal after we create a sample is to have a concentration between 20 to 50 micrograms per milliliter. And so as you can see, right here in this green box, the concentration is 46.8, which is just fine for us to store in a fridge to use later. Now, if the concentration was too high or too low, we would just take the solution back to the centrifuge and either centrifuge it more or just add some more buffer until we get the concentration that we want. Back in the lab, after we check the concentration of our nanostructures, we would take them and put them onto some type of substrate, let that deposit, clean it off, and then bring it down to our AFM room. So here's some examples. This one right here is a silicon wafer, and the rest of these are mica. Those have our DNA nanostructures on them already. And then next, we have to set up an AFM tip. So here's a box of these tips, fun fact. This whole box is $2,000. So we have to be very careful with how we handle them. That's why right here we've already got the tip set up in here. And once that's ready, we can take our sample and the tip to the machine. So how does the AFM machine actually take an image of our sample? Well, let's imagine my hand is our sample, and this is a DNA nanostructure. And so the tip that we talked about before would be lowered onto the sample, and once we start taking an image, it vibrates like this and moves along the sample. And once it gets to some sort of structure, it moves around it because it doesn't actually touch the sample, but just hovers right above it. So if there's anything in the way, it has to go above it. And that's how the instrument actually takes an image. And because the tip is so sensitive. Um, it's very sensitive to vibrations and sound, so it's in a fancy little box. And so we would take our sample and this, put our sample onto the glass slide here. Then we take our little thing with the tip and put it into this part of the machine to make sure it's secured. Then we take this very carefully, put it on top. And so now the wafer and the tip should be right on top of each other. And once this is set up, we go back to the software to set a few parameters. So after setting those parameters in the software here, we close the door to the machine, press go, and the image will show up in these boxes over here. And so this would be a 20 micrometer by 20 micrometer square picture. And once it shows us that, we can zoom in and out in different locations, which will end up looking something like this. So that was 20 micrometers. This is a 3 micrometer section. And here are our DNA origami triangles, which are just one of the structures that we're able to make. And so you can see this triangular shapes here, three straight sides, and you can see the holes in the center of the triangles. And what this this lets us know is that the structures were made correctly. However, you can also see some of them aren't quite perfectly triangular. Maybe some of them don't have a hole in the center, or maybe the sides just came apart. And that's what we're able to check in the AFM image.
Okay, so I also brought some actual samples from Pittsburgh. Now, those samples.
Hey guys, today I'm going to show you how we prepare our. This box is $2,000. And bring it down. So here's some examples. This one right.
Okay, so those are exactly the same samples she's using, and I'm just gonna show this around. Now, keep in mind, what is on this sample is tiny DNA structures. You can see them with atomic force microscope, but unfortunately, they're not visible to human eyes. Now, I wish I could bring the AFM here and show you everything, but before I become super rich, that's not going to happen. So there are two kinds of samples. One is a silicon wafer, that's a small piece. And then another sample is a mica glued onto a metal disc. The metal disc simply make it easier to handle the mica piece. So I'll just walk around, and if you're interested, you can take a look. I'm also going to walk in front of the camera so audience online can take a look. I'll give the mica sample to Dr. Kang, and he can show it around as well.
फोटो क्लिक करो, फोटो फोटो क्लिक किया था, क्लिक करना चाहिए ऑनलाइन दिखा।
I think that's pretty much what I want to talk about. Be happy to answer any questions. Um, any questions either about the current session or everything I have discussed in the past 5 days. It's been a
In the
great
past
pleasure.
5 days also, you can ask any question. Some students were asking which is the best substrate, whether it is silicon wafer or mica or maybe some other.
That's a great question. So both silicon and mica are quite often used. Mica usually, in my experience, usually give better quality of the image because mica is by its own, due to its own structure, atomically flat. Mica is a layered compound, and you can do exfoliation. It's also cheap. So mica has been a common and popular substrate for atomic force microscopy imaging for many years. We like silicon wafer for a lot of other reasons, in part because we've been developing lithography technique specifically for silicon. That's the reason we've been using silicon. But if you are just doing characterization of DNA structure, chances are you'll find better results on mica substrate. Any other questions?
Any other questions?
All right. If no more questions, again, thank you for your presence and attention to everything.
Please check if any online participant is having any query. We'll be starting the valedictory session shortly.
Recording in progress.
Good afternoon, everyone. First of all, I'll request all the dignitaries to please take your positions. Respected professors, invited guests, and dear participants, it gives me great pleasure to stand before you as we bring this enriching GIAN program titled Fascinating Features of Nanodimensional Smart Materials, organized by the Department of Industrial Chemistry, Aligarh Muslim University, to a close. This program has been a wonderful blend of insightful lectures, hands-on tutorials, and engaging interactions. Over the past 5 days, we have shared not just knowledge, but ideas, experiences, and a spirit of collaboration that will stay with us long after we leave this room. I would like to express a heartfelt gratitude to our esteemed Professor Mohammad Jangir Warsi sir, Professor Rafiuddin sir, and Professor Vibha ma'am for gracing us with their presence today. Also, I would like to extend my sincere thanks to our esteemed resource persons, Professor Hetaoliu, Professor Sahab Ali Askarnami, and Dr. Mohammad Zain Khan sir. Also, Professor Samir Sapra. Your expertise, patience, and dedication have made this course truly impactful. You didn't just deliver sessions, you sparked curiosity, challenged perspectives, and encouraged growth. To my fellow participants, it's been a pleasure learning alongside you. The discussions, questions, group work, and shared laughs created a warm and dynamic environment. Now, I would like to invite the course coordinator of the GIAN program, Dr. Mohammad Zain Khan, AMU, whose dedication and tireless efforts made this entire event possible. We are truly grateful for your vision and commitment, sir. We kindly invite you to present a report of the program.
Thank you, Aditi. It's really a pleasure to be with you all in the valedictory session of the GIAN program. Although I'm a little bit sad that we are now parting, we are closing this session. But the 5 days have been very enlightening for all of us. We have learned a lot. We have shared knowledge, experiences. I'm really thankful to the resource person, Professor Hitao Liu from the University of Pittsburgh, for traveling all the way from United States of America to India, particularly this point of time of the year, where the weather is also getting quite uncomfortable. I'm also thankful to Professor Samir Sapra. He came and delivered his lectures as well as tutorials on the 3rd day of the program. I really appreciate the initiative of the GIAN Global Network of Academic Network initiative of the Ministry of Human Resource Development, formerly called as Ministry of Education, which have started this program in which some eminent scientists around the globe have been invited to come to India and deliver lectures, teach our students, share their experiences. All the funds have been provided by the ministry. I'm really thankful to the coordinators of the GIAN program, the national coordinator, as well as the local coordinators. Fortunately, we have two local coordinators with us. We have Professor Jahangir Warsi Saab. He has been the local coordinator recently. He has moved to another university on an assignment over there. And now Professor Vibha Sharma, she is looking after the work of the local coordinator. And believe me, she is very cooperative and helpful throughout the organization of this program. We are very fortunate for the support extended by both the coordinators. I am really very thankful to the university authorities, the vice chancellors, and the other functionaries, and also to the auditorium and the in charge. In this 5 days program, we have organized 10 lectures, of which each lecture was of 1.5 hours, making it a 15-hour lecture session. Along with that, we have 5 tutorials of 2 hours each, around 10 hours of the tutorials, making it the learning process of 24 hours, which is equivalent to 2 credit course. That is the best thing that we have achieved. We have learned a lot in this week, in this 5 days program. Seeing the overwhelming response, we have stretched ourselves. We have got very good number of registrations. For this course, we have around 115 offline registrations, and online registrations were 70, making it 185 enrollment of the students. Please give a huge round of applause, students. Out of which we have 25 research scholars, around 15 faculty members, more than 50 UG students, and around 70 PG students. As part of this course, we have also organized a visit to the university's sophisticated instrumentation facility, as well as the Interdisciplinary Nanotechnology Center, because a big weightage in this course has been given to the skills that student can learn throughout this course. And for that, we have arranged this visit, and I'm really very thankful to the coordinators as well as the directors of those centers. Their team was very efficient, and they explained 5 advanced equipments. They gave demonstration to the students: the X-ray diffractometry, the single crystal X-ray diffractometry, then the transmission electron microscopy worth of crores of rupees, the scanning electron microscopy, and the Raman spectroscopy. These 5 instrumentations we have covered during the visit. Now, as part of fulfilling the mandatory criteria of this GIAN course, 3 things are necessary. First is the attendance. We have maintained the attendance in each and every sessions. For the online participants also, we have maintained the attendance. After the attendance, the next thing is that an assignment should be given to the participants. All the students have successfully submitted their assignments with the support from all the faculty members of the department. They worked very hard, and last night only we have covered the evaluation process. The students performed very well. And today we have the third requirement was an examination based on multiple choice questions that has recently been held from 2 to 3 p.m. The copies are still under evaluation by the faculty members. So we have completed those requirements. Students have worked very hard. The participants worked very hard. In addition, we have also organized a quiz competition based on some online questions relating to the topic of the GIAN course, and we have distributed prizes to the winners of that program. Then there was an expert lecture on one of the very latest computational technique known as density functional theory for studying the structure of molecules and their interaction in a composite. The lecture was delivered by Dr. Deepak Kumar from Quantum ATK company, from Gurgaon. This is all about the activities that has been performed during the last 5 days. I once again thanks all the participants also, because it is because of you the event has reached to its zenith. With this, I am now closing my words, and I am really thankful to all of you. Thank you very much.
Thank you so much, sir, for your kind words and for being the driving force behind this successful program. Your leadership, passion, and meticulous planning truly made this week a memorable and enriching experience for all of us. Now, it is my pleasure to invite the esteemed chairman of the Department of Industrial Chemistry, Dr. Shahab Ali Asghar Nami, to share a few words with us and offer his insights on this memorable occasion. Please, sir.
Chief guest of this 5-day GIAN program, Professor Hitao Liu from University of Pittsburgh, USA. Guest of honor, Professor Samir Sapra from IIT Delhi. Today's chief guest, one of our celebrated and very loving teacher, Professor Rafiuddin Saab, DSW. Local GIAN coordinators, both past and present, Professor Varsi Saab and Professor Vibha ma'am respectively. Course coordinator, Dr. Muhammad Zain Khan. Esteemed faculty members of the dais, colleagues, and my dear students. As Dr. Zain Khan has said, it's a sort of mixed feeling. Of course, a feeling of satisfaction and a feeling of going apart after this 5-day thrilling experience where we have learned together, where we have shared the techniques, we have shared the live experience. And I'm particularly thankful to Professor Hitao, who came a long way from USA in this, in the scorching heat. And he delivered very insightful lectures. And he also takes some samples with him, where he shared those samples, those live samples with you, to just have a clear-cut idea, to have a living experience that how these nanostructures look like from the naked eyes. Although you cannot visualize how much DNA is fabricated on it. But of course, this gives you an idea that how the science is going on and how far actually the science has gone. So, during this 5-day program, I understand that you have experienced certain issues, and with regard to your stay, with regard to your food, with regard to your travel from one place to another. But I think that the foremost aim of this program is to gather knowledge, is the enrichment, and to make you a visionary, because someone has said very right, an idea can change your life. So you have to have a quest of knowledge, and the aim of this GIAN program is to empower you with the latest developments, not on an Indian perspective, but globally, to give you a platform where you can interact, where you can share, where you can discuss, where you can collaborate with the foreign experts. And of course, Professor Hitao Liu is one of the shining stars as far as the DNA nuclear chemistry is concerned. So I give a very big hand to Professor Hitao. Now, since all of us know that we are a very new department, actually we are a department which is still in its infancy. And today, that is on 18th April, we are only 2 years, 3 months, and 17 days old. and we understand that being younger is sometimes very good, because if we have, अगर कहीं कमी रह गई तो उम्मीद है कि लोग दरगुजर करेंगे. So this is like that. Now with regard to the GIAN program, it is a wonderful initiative by the Government of India, by MHRD, who started this program to get an idea, to give you a platform where you can have a hands-on recent knowledge of research particularly. This idea is to get you fortified with the latest developments. So I'm sure that you have a fruitful discussion, you have fruitful learning. And at the last, I want to pay my sincere gratitude to our Vice Chancellor ma'am, to our Vice Chancellor sir, to the Dean Faculty of Science. All of us are not here with us, unfortunately. And to our DSW sir. He is also one of my teacher and celebrated teacher, a very loving teacher, Professor Rafiuddin Saab. So give a big hand for joining us. I am also grateful to other faculty members, notably one of the leading plastic surgeon, Professor Imran Ahmed Saab, who also spared time and graced this occasion. So in the end, I want to say that we are leaving with the hope that we will, come someday again and share the vision and the beauty of science. So with these words, I'm thankful to all of you for joining us. And if you feel any difficulty, so kindly ignore that and have a fruitful learning, and good luck in your future. Thank you.
Thank you, sir, for your thoughtful words and constant support. We truly appreciate your guidance and encouragement throughout this program. Now, it is my distinct pleasure to invite someone whose presence and support have added great value to this program, Professor Vibha Sharma, Local Course Coordinator, GIAN. Professor Sharma's dedication to academic excellence and her unwavering encouragement for initiatives like this program have played a key role in its success. May I now request Professor Vibha Sharma to kindly come forward and say a few words.
A very good afternoon to all of you. Assalamu alaikum. It is indeed a pleasure for me to be standing here at the juncture of the closure of this course, which is Industrial Chemistry Department's first GIAN course, and I'm sure it is not the last course, because the way you are going, I'm sure that you will have many more such courses to boast about in the future to come. And I would have preferred, or maybe it will follow after my speech, some responses from the participants, if you permit, because that will check the other box for me, like, participants' response was also good. And of course, we are going to listen to Professor Haitao Liu. So all the esteemed guests who are sitting here are not guests. We are a team. Professor Rafiuddin, he is going to preside the session. He is also Dean Students of Welfare. And Professor Haitao Liu from Pittsburgh University, Professor Jahangir Varsi, whose work I'm taking care of in his absence. This is his work, and I'm simply here to take the credit, so which is not correct. So I must share the credit for the hard work that he had been doing for these courses to get incepted when they were applied for. And the young team of this department, Dr. Shahabnami and Dr. Zain, two very dynamic teachers of this department. So it is indeed a very, very pleasant moment for all of us. As teachers of one university, we feel proud that any part of our university achieves something. And this course is a very interesting thing because the word nanotechnology figures in this year's season because of this course. This is a word to go for. I am not anybody to speak about it, but the future generations are going to develop only because of these kinds of sciences that are around the corner in every part of the world. Now, I must also say that the way this course has been handled very efficiently, Dr. Zen's hard work must be praised for. I think he deserves a big round of applause. At this stage of career, it is not easy to pull through such wonders, but he has done that, and it is indeed a thing worthy of big praise. When Dr. Nami was talking about the young age of the department, I am reminded of something, that this is the inception and this is the formative stage, like a child's infancy. And in infancy, the personalities are shaped. So the department's personality and future vision will depend a lot on the actions that are being taken up by you people at this moment. Not just the teachers, but the students. Maybe you are not realizing, but I must signify as much as I can that the presence of the students and what you are bringing to this department on the table will be the future direction for this department, which is now going to become a bigger place in years to come. So participate more and more, give your best to this department, to this university, and I think you should shine brighter in the sky of achievements globally, because India is not going to achieve anything unless its young scientists are able to transform them into world scientists, global scientists, and achievers for the world. So my best wishes for that. And I must, not just formally, but from the bottom of my heart, thank the university administration for always being very supportive, led by none other than but Professor Naima Khatun, the Vice Chancellor, and her dynamic team. And I'm very indebted to our government's visionary scheme, that is Global Initiative of Academic Networks, that stands before you in the acronym called GIAN, which is a Sanskrit word also. So let the light of GIAN prevail in our hearts and mind, and let this be the force with which we move ahead. See you in other GIAN courses very soon. Thank you.
Thank you, ma'am, for your thoughtful words and constant support. It is my distinct honor to invite our guest of honor, Professor Mohammad Jahangir Warsi, sir. We are truly privileged to have him with us today.
The chief guest for this evening, Professor Rafiuddin Saab, the Dean of Students' Welfare. Professor Tao Liu, just I came to know that when I told him that I was at UC Berkeley, he immediately said I did my PhD from the same university. So thank you for being here for this meeting. Professor Shahab Ali Asgar Nami Sahab, the chairman of the department. Professor Vibha Sharma, the course coordinator, Dr. Zan, all the learned colleagues, all the participants, and my dear students. A very good afternoon and assalamu alaikum. I am very thankful to Zan, and I must congratulate him and the department for having this GIAN program successfully conducted here today. A GIAN program, like since I was the earlier course, local coordinator of the university, but the hard work for this phase, whatever it has been done, all the course coordinators deserve our appreciation, and so as the local coordinator, Professor Vibha Sharma. You know, there are so many communication that is needed to coordinate with the national coordination. Unless you have that kind of caliber to coordinate, to interact, to answer, to send the required information, it is very like so much technical bureaucratic hurdles, and we have to overcome to those to have a successful GIAN course like this. So I must congratulate the Zen, the chairperson, the industrial chemistry department, and overall the faculty that they belongs to. As all the people have said, like this is not anything that you are in the classroom. This is whatever you are learning. Most of you are undergraduate, postgraduate students. Whatever you are learning, we are giving. This GIAN course just give you a perspective what is happening at the latest in that area of expertise. And this is like the government of India spending so much on this GIAN program, that providing you the expertise, the best of the minds of the world in globally available for you to come here and deliver the lecture, along with the tutorials that you have had during these 5 days of the successful GIAN courses. This is going to 100% going to enrich your understanding what is latest happening in that area and how you are going to carry this rich knowledge that you are getting from this expert, from these lectures. And I was reading a news report from the inaugural session, and I was happy to see that the experts, they are the leading names in their respective fields. So I must congratulate the entire team of the Industrial Chemistry Department for organizing this successful GIAN course. These are the things that you, as Professor Vibha Sharma rightly said, the light, the GIAN that you are getting now, you have to let all throughout the globe when you become the alumni of this university, when you graduate from this university. These memories will always remain throughout your life, and you are going with this knowledge to disperse, to de-embark from all over the world. Like wherever you go, you will remember, you will cherish this time that you are spending here, getting the knowledge from the leading teachers, from the experts, from everyone. I must also congratulate especially Professor Vibha Sharma, that I was in constant touch with her. So many things that we have to coordinate, कि ये कैसे होगा, इसको कैसे जवाब देना है. And one of the, that I could not do during the tenure that she has done, one of the GIAN course that the expert, like, these GIAN courses are very specific expertise that GIAN implementation committee chooses or select. One of the GIAN course, being like the expert chosen for the course, was refused to come to India because of the health reason. And the course coordinator, he or she was very upset, like how I'm going to conduct this course. Because of Vibha Sharma's communication with the National Coordination Committee, they have agreed to change the expert for the same course, and we are going to have that sometime in July. So credit goes to you, Vibha Sharma. These are the occasions we get to know internationally, like the expert, the opinion from the different persons. We may disagree on some aspect, but we always enrich our understanding. We are always going to improve our understanding and knowledge of the subject. So with this, most of you are students and have learned all the many things from his expertise. I do not belong to the science. My area is linguistics and languages, so I have nothing to say about the expertise, but what I understand from the topic, that the world is going towards interdisciplinary and multidisciplinary approach. We cannot remain confined to one aspect. So develop your understanding, how you can be interdisciplinary in your approach, how you can be multidisciplinary in your approach to make your career, to build your career, and that will help you in the future to come. And since most of you are a student, I will end up by saying, खुदा करे कि तेरी जिंदगी के गुलशन में, खुदा करे कि तेरी जिंदगी के गुलशन में खिजान आए, सदा मौसम बहार रहे. बहुत बहुत शुक्रिया. Thank you very much.
Thank you, sir, for your kind and encouraging words. Now, we'd be honored to hear a few reflections from the minds that led this program. May I kindly invite our chief guest, Professor Hitao, to share a few words with us.
First of all, I have to express my sincere gratitude for everybody behind the scene who made my visit to this event possible. It is my first time visiting India, and I have benefited from a lot of collaboration, mentorship from Indian colleagues, contribution to my research program from Indian students and postdocs. I just felt I just started to repay a little bit of that of all the benefits and mentorship I have received. I have been working with Dr. Kant for a number of years and interact with some AMU students online. but this is my first time to meet with so many students and faculty in person, and I just felt it's an incredible experience for me to know everybody at a personal level. The GIAN program is such an incredible program. I think the Indian government is doing a great thing to spend money on education and educating students by bringing experts from all over the world to have face-to-face time with students and faculty. I wish there could be more opportunities like this, not only in India, but also in other countries. This 5 days is, as strange as it may sound, is an incredible educational experience for myself. I got the chance to really refresh my own scholarship and academic learning. People usually see the best way to learn something is to teach something. And I'm certainly doing a lot of learning in the past 5 days. All the questions you have thrown at me, I feel very fortunate to hear about your thoughts. Those are questions that stimulate not only your own thinking, but also mine. And I really appreciate the opportunity to exchange scholarship with students and faculty here. And for that, I certainly would thank the Indian government for providing the funding, all the faculty and staff behind the scene to make this possible. I know this requires not only money, but also human interaction and care and engagement. This is such an incredible institution with not only outstanding students, but also outstanding faculty and administration. So for that, I feel so fortunate to have experienced India in this way. And last but not least, thank you for all the student organizers, volunteers, and everybody who participated. As I said, you gave me a wonderful learning experience, and I hope I have repaid some of that debt, and hopefully some of my lectures you'll find useful in the future years. I'd really happy to continue the discussion over email or other means. If you have any questions, research questions or otherwise, I'd be happy to continue the discussion in email exchange. And again, thank you very much. And it's an incredible experience for me. I hope this will be a useful experience for you. Thank you.
Professor Hetaulu will be in the Department of Industrial Chemistry tomorrow. So if anybody wants to interact with him regarding the graduate program at the University of Pittsburgh, he or she is most welcome. By 12 noon or 12:30 p.m., he'll be in the department. Thank you.
Thank you so much, sir, for your inspiring words and for gracing this occasion with your presence. Your insights have truly added depth and meaning to this GIAN program, and we are grateful for your time and encouragement. Now, I would like to invite Professor Rafiuddin, Dean Students Welfare, Aligarh Muslim University, for presidential remarks.
Thank you very much. Professor Hetao Liu from University of Pittsburgh, United States. Dr. Shahab Nami, Chairperson, Department of Industrial Chemistry. Professor Vibha Sharma, Coordinator, GIAN. Professor Mohammad Warsi Sahab, now perhaps Director Academic Program in Jammu and Kashmir. Dr. Mohammad Zain, all the teachers who are present here, Professor Imran Ahmed, other teachers, colleagues, my dear students, ladies and gentlemen. It is truly a very emotional moment for me when I'm speaking for Industrial Chemistry's sponsored program on nano-dimensional smart materials. My relation with industrial chemistry, of course, goes back to many years when I was coordinator of industrial chemistry for 5 years. And that was the time when I used to arrange teachers from mathematics, engineering college, commerce, management, from almost every other faculty. We try to get teachers who can teach students of industrial chemistry. Initially, it was a vocational program, and gradually it came into the mainstream. So those time, I, as a coordinator, I tried my best, and in my time, I passed the resolution in BUS to have this separate department. And now, when it was passed, I was in court also, in academic council, and it's really very exciting moment when you see yourself that a new department has come up, which has a great potential, which has a great scope, and luckily, the department has got teacher like Shahab Nami, Dr. Zain. These two, along with, of course, other teachers, these two are trying their best against all odds to develop this into a major department. And I'm sure in very few years, it will be a full-fledged big department. It's full-fledged department, but it will have as far as this program of 5 days is concerned, as I was told by the organizers, by the chairperson, by Zain, that nearly 170 participants took part in this program, which was lectured by very learned people, including Professor Samir Sapra and Professor Etau Liu, along with other teachers. I'm sure all of you would have learned many information about nanoparticles, nanoscience, nanotechnology, along with certain instrumentation and instrumental approach. One thing as a teacher of chemistry, I must tell you that whenever you plan a new field, you must be clear about fundamentals. There is no substitute for fundamentals. Unless you have a good understanding of the subject, you cannot understand the advances in the new field. So for understanding advances, for understanding nanotechnology, nanoscience, and why nanoparticles are so important, you have to have a good understanding of mathematics, physics, and chemistry. You are working on DNA, but unless you understand the basics of chemistry, it will not be possible. You see, the Nobel Prizes are awarded not in medicine, but for medicine they are awarded in chemistry or physics or some other subject, because to create new knowledge, you need to have understanding of the fundamental of subject. In the recent time, you see, even the subject like biochemistry, you have lot of people from mathematics background. In the very beginning, people were never thinking of that. But without mathematics, without physics, you can never have a good understanding of the subject. So my advice to you, as you are a student and you are going to do some good things in this field of nanoscience, that you try to have a good understanding of the fundamental of the subject, then only you can do good work in this field. And the field changed. You see, if you go to the history of the science, and if you look at the CV of good researchers, you'll find after almost every 10 years they change their field. No one remain in one field for 40 years because that become obsolete. So to do the things in right direction, you need to have a good understanding of the subject, and thereafter you go for the advances. So I'm sure by attending such workshops, such seminars, lectures, and interacting with people like Hetao Liu, Samir Sapra, and your teachers, you get lot of understanding of subject. So I congratulate the organizers for organizing such a wonderful program. The chairperson, Dr. Shahab Nami, Dr. Mohammad Zain, and GIAN local coordinator, Professor Vibha Sharma, who has been, of course, instrumental. And for me, certainly I feel more happy because she is also associated in my assignment as Deputy DSW. She is helping our job, our task. She is doing so many things. Coordinator of GIAN, MIC, public relation, along with a good English teacher. Professor Varsi Sahab is, of course, doing so many assignment and so many responsibility. So this is the thing in the system of the university, that a teacher is supposed to do many things, but the first thing is you need to be a good teacher, you need to be a good researcher. Unless you are that, you will not be able to do other things. So again, I am thankful to every one of you, and particularly the organizer and the chairperson and Mohammad Zain for inviting me on such occasion. Thank you very much.
Thank you so much, sir, for your kind and encouraging words. Now, I would like to invite anyone from the audience to come up on the stage and share the feedback for the GIAN program.
मेरा नाम धर्मेंद्र सिंह है, मैं कासगंज से आया हूं और लास्ट ईयर बीएआरसी का जॉब छोड़ा था मैंने, लाइब्रेरी टेक्नीशियन। डाइस पर उपस्थित सभी सर और आप सभी लोगों को मेरा नमस्कार। तो सबसे पहले मैंने एक कविता लिखी है ज्ञान के ऊपर, तो वो सुनाना चाहता हूँ। काश सीखने सिखाने का ये दौर ए सफ़र कभी ख़त्म ना हो, काश सीखने सिखाने का ये दौर ए सफ़र कभी ख़त्म ना हो, और वक्त ठहर जाए ताकि वक्त का पता न चले और ज्ञान का नवसृजन हो सके। तो ये कविता मैंने ज्ञान का एक NITJ में अटेंड किया था वर्कशॉप। वो था मतलब कंप्यूटर से मैथमेटिक्स से जो इमेज बनाते हैं उस टॉपिक पे था, तो तब लिखा था। इस प्रोग्राम को अटेंड करके मेरे को काफ़ी अच्छी नॉलेज मिली, और मैं BARC मुंबई में जब इलेक्ट्रोकेमिस्ट्री में एक वर्कशॉप हुआ था, तो उसमें फ्यूल सेल और हाइड्रोजन स्टोरेज वगैरह के बारे में बताया। तब से मतलब केमिस्ट्री की तरफ भी मेरा थोड़ा रुझान बढ़ा। वैसे मैं मैथमेटिक्स का स्टूडेंट रहा हूँ अच्छी तरह, तो मैं चाहता हूँ कि आगे मतलब ऐसे ही वर्कशॉप अटेंड करते हुए कुछ नया चीज़ सीखने को मिले। यहाँ पर काफ़ी अच्छी चीज़ें सीखने को मिली। ग्रेफिन के बारे में जानकारी मिली, नैनो ट्यूब के बारे में जानकारी मिली, और क्या बोलते हैं उसको, ये DNA वगैरह स्ट्रक्चर वाला सर ने जो बताया था लास्ट लेक्चर, वो कम समझ में आया लेकिन काफ़ी अच्छा नॉलेज मिला। तो थैंक यू सर।
Thank you, Dharmendra ji. Thank you.
Thank you very much, sir. Now, I would like to invite Ms. Ruhi Siddiqui to share her feedback.
Good afternoon and Assalamu alaikum everyone. Myself Ruhi Siddiqui and pursuing MTech from Center for Advanced Study, AKTU Lucknow. So it's a great honor to share my experience as a participant in GIAN workshop, which is on fascinating features of nanodimensional smart materials. So which is organized by Department of Industrial Chemistry, Aligarh Muslim University. Throughout my 5 days workshop, I had the privilege of learning from world-class experts, Professor Hitaw Liu, who introduced us to synthesis, interfacial properties, and versatile applications of 2D and carbon nanomaterials. Dr. Mohammad Zain sir, who guided us through 2D nanomaterial and instrumental techniques like cyclic voltammetry, electrode fabrication techniques, and others. So this electrochemical analysis and hands-on demonstrations like XRD, TAM, SAM, and Raman spectroscopy was very helpful and understanding the characterization techniques. Dr. Shahab Ali Asghar Nami sir, who shared cutting-edge research on nanohydrogen gels in soft robotics and wearable devices. So beside of this, I must appreciate the hospitality and arrangements at AMU. The guest room was so comfortable and the food was so good. So everything was well organized, making this experience smooth and memorable for me. So thanks to all the professors, coordinators, and the AMU staffs for all the valuable support and time. Thank you so much.
First of all, Assalamu alaikum wa rahmatullahi wa barakatuh. Good afternoon, everyone. I, Anam Fatima, research scholar from GB Pant University of Agriculture and Technology, Pantnagar, Uttarakhand. First of all, I am highly thankful to the coordinator of this program who gave an opportunity to attend this GIAN workshop, which was based on fascinating feature of nanodimensional smart material. And now I am thankful to committee members and organizer who are involved in this workshop for going smoothly. Now I have shared my experience which was I gained from this workshop. This workshop is very informative and very well informative. And I have learned about many things about 2D nanomaterial, which lecture was given by Professor Liu. And now I have learned many more electrochemistry technique such as voltammetry, cyclic voltammetry, linear sweep voltammetry, which was taught by Dr. Mohammad Zain sir, which was very informative from my research perception. Now I would like to the lecture of Dr. Mohammad Shahab Ali Asghar Nami sir, who gave the lecture on the polymer-based hydrogel, which is also useful because my research was almost similar to that material. Now, I have in this session we have gained many theoretical and practical knowledges and some software tools which are using in this technique. Now I have the session was very good and very smooth. Now the hands-on training session, which is very helpful and very informative, and I am exciting to learn how to these smart material can be used in recent advancement and research techniques. And thank you once again for this valuable learning session. Thank you so much.
Else for the feedback from AMU students, if they want to share their experience, they are most welcome. Anybody?
Now, I would request Dr. Shahab Ali Asghar Nami sir to present a folder to Professor Vibha Sharma ma'am. I would request Dr. Mohammad Zain Khan, sir, to present a folder to Professor Jahangir Warsi, sir. I would request Dr. Shahab Alias Garnami, sir, to present a folder to Professor Rafiuddin, sir. Now, I would request Dr. Mohammad Zain Khan, sir, to present a folder to Hitao, sir. Thank you very much, sir. I now invite Dr. Shoaib Akhtar, sir, to deliver the vote of thanks and express a heartfelt gratitude to everyone who made this program a success.
Good evening, everyone. As we come to the close of this intellectually stimulating and fruitful GIAN 1 week program, it is my privilege to deliver to vote of thanks on the behalf of the Department of Industrial Chemistry and the organizing team. First and foremost, I would like to express our deepest gratitude to our Honorable Vice Chancellor, Professor Naima Khatun ma'am, for his constant encouragement and unwavering support. Your vision continues to inspire academic excellence and innovation in all our pursuits. We are also immensely grateful to our Pro-Vice Chancellor, Professor Muhammad Mohsin Khan sir, for his constant encouragement and for always being a pillar of strength in all academic endeavors. A very special and heartfelt thanks goes to our distinguished chief guest, Professor Hetao Liu from the University of Pittsburgh, USA. Sir, your sessions were intellectually stimulating and have certainly broadened our academic horizons. Your expertise and generous engagement have left a lasting impact on every participant. A sincere thanks to our guest of honor, Professor Samir Sapra from IIT Delhi, for taking out your precious time to join us. Your contribution to the scientific community is remarkable, and your presence has truly encouraging for young participants. It is also a privilege for me to deliver the vote of thanks to our esteemed chief guest for today's valedictory function, Professor Rafiuddin sir, Dean Student Welfare, Aligarh Muslim University, Aligarh. Sir, we are truly honored by our gracious presence and your thoughtful address that added great value to his concluding session. Your words have not only inspired us, but have also encouraged deeper reflection on the academic and research path we pursue. I would also like to thank our Dean Faculty of Science, Professor Sartaj Tabassum sir, whose proactive leadership and constant support were crucial to the successful execution of this program. It is my sincere honor to extend a special vote of thanks to Professor Vibha Sharma ma'am, our local GIAN coordinator, Aligarh Muslim University, Aligarh. Ma'am, your dedication, meticulous planning, and unwavering support have been backbone of this entire program. I would also thanks to Professor Muhammad Jahangir Warsi sir, ex-local GIAN coordinator and presently Dean Academic Affairs, Baba Ghulam Shah Bahadur Shah, University Kashmir, for joining this program and encouraging the young participants. I would like thanks to Professor Faiza Basim, Director of UGC HRDC AMU, for providing the necessary facilities and wholehearted support to ensure the smooth running of this week-long academic journey. I would also like to warmly thank our course coordinator, Dr. Muhammad Zain Khan, for organizing such a wonderful GIAN program, who tirelessly worked day and night to make the program successful. And our course coordinator and the chairman, Department of Industrial Chemistry, Dr. Shahab Ali Asghar Nami, for his relentless efforts and leadership throughout the planning and execution. We are deeply thankful to the MHRD, Government of India, for providing the necessary funding and for launching this GIAN 1 week program that has truly opened doors to international collaboration, cutting-edge knowledge exchange, and academic enrichment. A special mention must be made of our esteemed team members, Dr. Abu Mustafa Khan, Dr. Rizwan Hussain, Dr. Muhammad Asfar Sheda, Dr. Muhammad Tariq, and Dr. Farasha Sama, for their consistent commitment and contributions, both seen and behind the scene. Last but not least, this vote of thanks could not be completed without giving thanks to our dedicated young research scholar team, including Mr. Waris, Mr. Suhail, Miss Ariba Khayal, Miss Aditi Agrawal, Miss Shumaila Hassan. Their day and night efforts make run this program very smoothly. To all dignitaries on the dais and off the dais, we thank you with great respect and appreciation. Lastly, a warm thanks to all participants, faculty members, technical staff, and students who have joined us today and are contributing to the success of this program. With heartfelt gratitude, thank you all once again.
We are very thankful to Dr. Shoaib Akhtar also, who has been in charge of the food committee. Because of his efforts only, we have got very delicious food for the last 5 days. A special thanks to Professor Rafiuddin sir. He has always been very supportive to us, not only for today, but since the beginning of this department and much before the beginning of this department when we were in the Department of Chemistry. He has always been very supportive and encouraging. A huge round of applause for the Mr. Scholar teams who has been, Baris, come here. Ariba. Very, very well. They have worked very hard. Fahad, you also. Zaim also, and all those who have been working very hard, Aisha, and to our faculty members like Dr. Rakshanda Rahman, Dr. Nimra Shakil, Dr. Tuba Fatma, Dr. Rabia. I am really very thankful to you for your support during the evaluation process of the assignment as well as the examination based on MCQ. Thank you. Time for AMU Tarana and National Anthem. Participants, we should have another program next year maybe. What do you think? GIAN program on some related topic? Sure, we'll try our best.
मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे.
The valedictory session of the GIAN Global Initiative of Academic Network course has closes now. I request everybody to please proceed for a cup of tea.